Okay, hello everybody. I hope you can hear me. If you're coming in, just put a one in the chat room. If you can hear me, I'm going to go ahead and post the link if you want to come up and join me. But in the meantime, I'm going to put the PowerPoint on uh, full screen. I'm going to see if I can hear myself on my phone. One moment. Let's see here. Yep, I can hear myself. So you all should be able to hear me just fine. I'm going to put this on full screen. Let's see. How you doing, Robin the Girl Wonder? How are you? Let me go to full screen so you all can see it. And this is just going to be a quick, hopefully, excuse me, quick discussion as a follow-up to Dr. Dunbar's work that he did on Saturday. I was on that stream. I have a link to it in the description box. If you will, as you're coming in, please hit that like button. If you have not subscribed, please hit that subscribe button as well. You can support me at Cashapp below, also by just hitting a like, giving your comments as we go through this PowerPoint. So I prepared a PowerPoint that was going to be pertaining to Dr. Dunbar's discussion that he was having on Saturday. I'm so grateful that he invited me to come up, and I hope that you all learned something from Saturday's discussion. It was 3 p.m. Pacific, 6 p.m. Eastern. We had a good crowd in the chat room. And so the title was What We Know About the Science of the Coronavirus COVID-19 Pandemic. Please hit a one in the chat to let me know that you can hear me okay. And I'm going to keep going. So let's see here. All right. I'm trying to get over whatever this little issue I'm having. Uh, I'm going to blow it up to full screen and I'm going to keep it going. Black Uru Strikes, how are you doing? Chaos Rain, what's going on? Robin, I said hello to you before. Thank you for coming, everybody. This is impromptu, but I hope that it's okay that I'm doing this. Of course, you all, if you don't have the opportunity to watch it right now, you can always watch it later. I just appreciate the likes and the support that Dr. Dunbar is getting. He absolutely deserves it. And once again, I thank him for allowing me on his channel the other day. So, Big Discussion 76, Science and Technology with Dr. Anwar Dunbar. And I'm going to go into a lot of information that I didn't get a chance to have there, which I wasn't expecting. I just like doing PowerPoints, y'all. I'm just going to be honest. So, I thank you for the ones in the chat as well. Oh, there's Dr. Dunbar now. How you doing? You said taking a break from the madness here. I feel you on that. Definitely. But I wouldn't say that like it was expected I would give a presentation. I just like putting presentations together. So now I have an opportunity to do that here. And his channel, he wants to do things, the way he does it. And I appreciate that. And I just wanted to put forth links and more information for you all. If you want a copy of this PowerPoint presentation, please email me at the email address below. Your screen is highlighted in blue or like a turquoise color. Excuse me. Like I said, I'm trying to deal with whatever it is I'm dealing with right now. But this is my lab station on any given day. I was doing, I think, real-time PCR. So I have a lot of pipette tips. I have PBS here, some buffers for washing. I have some PCR plates here, some notes in my notebook, calculations, pipettes, tips, tubes, more tubes here and everything, more plates, tubes, racks, the whole nine. So what I wanted to say here was a messy workspace doesn't always equate to a messy mind. We have to remember that we're all a work in progress. I have more than 10 years research experience in biomedical field. I began with microbiology, immunology, but I quickly transitioned into cancer genetics. specifically prostate cancer genetics, and I've been doing that for the past 4 years with gene therapy and engineering. My primary targets, as you know, are prostate cancer, that is specifically metastatic prostate cancer. I focus on genes that promote prostate cancer and interact with the genes that interact between two different cell types. One is epithelial prostate cancer cells, and the other is fibroblast prostate cancer cells. My primary models include prostate cancer cells, tissue. Well, these are cells that are directly from the prostate, known as primary cells. Also, prostate cancer cells you can buy from ATCC for our laboratory, tissues, blood, plasma from patients, or tissues, blood, plasma from mice. We also genetically modify our own cells. What's going on, AB Media? India XO, how are you doing? Oh, thank you. Thank you. Thank you for being here. So I want to remind everyone that SARS and the study of this family of viruses is not new. I repeat, it is not new. I wanted to also provide a timeline of the development of this virus, how it was identified, studied, and everything that they're doing up until now. So in red, I have SARS-2 novel coronavirus 19, aka COVID-19. It is novel, meaning it is new to public knowledge. It is not new to research, but it is new to public knowledge. And I have a timeline here just to give you an idea of the development of these particular viruses and how things have, come about. So the human version of influenza A, H5N1, is shown in from this paper to be related to a highly pathogenic avian influenza bird flu. That was in 1998. Moving forward into 2000, 2003, all the way up to 2008, you see the way that these viruses are manipulated and being used for further modeling, gene targeting, understanding the genetics, the complementary DNA strands, and identifying and studying SARS in different models. So this is actually a longer list of publications. I've just given 3 that I'm really interested in right here. And finally, coming up to 2020, we have this pandemic that we're in right now. Many reports have not been fully published. So the authors are using bioRxiv to put their papers out, at least on the internet, before they can be peer reviewed, because peer reviewed publications, excuse me, do take time to put out. Currently, there are more than 3,400 clinical trials in progress for vaccines of this virus. And as we know, we talked about those in detail on Dr. Dunbar's channel last Saturday. Okay, so I have a slide here about viral studies conducted from late 1990s to the mid 2000s. And I've highlighted 4 of them because these also relate to my timeline. Okay. And the others also relate to the timeline, but not as specifically. So many of the authors are virologists, microbiologists, immunologists, because they're dealing with viruses and also dealing with the pathogenicity of these viruses. And what you see in many of the papers is they're trying to isolate the genetic sequences of these viruses and characterize them in specific demographics of patients and specific geographic areas, so mostly in Asia. So one paper that I pulled out, I wanted to discuss a little bit further, is the SARS coronavirus genome, and showing that it does not match the human genome. And this study was performed in 2003. So the title was Isolation and Characterization of Viruses Related to the SARS Coronavirus from Animals in Southern China. And what they find is that they detected a similar COVID-like virus in small, live, wild mammals in a retail market, and indicated a route of interspecies transmission although the natural reservoir at that time was not known. And they give here different detection of antibodies against the recombinant nucleocapsid protein of COVID. So they have the different recombinant capsid expression or sequences here. They have the human sequence as a control, and they have that there is some sequence identification or homology, but not for all of the sequences to human. So here's the human band at 50 kilodalton, and there are a few of them that do have similar expression at 50 kilodalton, but the concentration of the antibody or the amount of antibody there is not as high as it is in human. Then they went and showed phylogenetic analysis to determine if there was a similarity in sequences or mutations between humans and animals. And there are some similar mutations within humans, within the human population, but there's really not much sequence identity or similarity between humans and the animals that they chose to look at here in this study. Furthermore, they go and show that the open reading frame 1A, open reading frame 1B, and several of the downstream markers for SARS-CoV-19, or excuse me, not COVID-19, but SARS coronavirus genome does not match that human genome by matching human to animal genome. And they notice that this red sequence here is not found in humans, but it's found in several of the animals. So when trying to determine if humans, excuse me, who were infected with SARS got it from an animal species, all they would have to do was determine if this red sequence right here is in the humans, is in the human specimen that they're analyzing. If so, then yeah, it has jumped from animal to human, and that's how they would determine that. So I'm not sure if people know, but the genome DNA sequence that you're looking for is composed of cytosine or C, thymine T, adenine A, and guanine, which is the G. So ATGC, right? And the protein sequences are all different letters: A, L, G, K, V, Y, Q, H, all these different letters. These are protein sequences. So I also put their figure legend to help with understanding what they're talking about here. In the A, they have a deletion, as they say, 29 nucleotide deletion in human. They have a deletion, or that's not seen for animals, for instance. And then they expanded that out. And then with C, with the protein sequence, there's not much alignment. So open reading frame 10 and 11 are human, and 10 prime is animal. So 10 and 11 are human. They're not very similar, not too much. But this animal one, seems to be complete. So there is a deletion somewhere here at least for one of the human samples that was taken. So there's a deletion here for ORF10 and here for ORF10. So this protein is very short. And a deletion here, ORF11, which is also human. And this is where the protein sequence starts here and goes here, all the way to 122 amino acids. And then open reading frame 10 prime is an animal isolate, and it is complete. So there's things that the animal sequence has at the DNA level and the protein level that humans don't necessarily have. So that's where they recognize that there was a genome mismatch between humans and animals. So let me look at the chat real quick. We got Torian Rain Reloaded. How are you doing, Torian? Herbert, how you doing? AM1, Des said anime. AM1 said it almost sounds like they're implying that it was engineered. So this is the first, this is natural form of SARS coronavirus from animals. And what they were doing in 2003 was to determine, they were trying to determine if there is sequence homology. Let me go back. If the human and animal sequences for the genes responsible for the spike protein expression, if they were similar, and they find they're not similar. And then in between humans, open reading frame 10 and 11 are two human. So even within humans, there's a difference. And this also equates to, or it leads to the idea, implies that humans, of course, we know have differences in immunology. Dead set, he said, wow, I learned something every time I visit this channel. Oh, thank you. I appreciate that. What's going on, Nameless? He says, salute to the chat, respect to the doctor. Yes, respect to Dr. Anwar Dunbar. Thank you again for having me on your channel on Saturday. I appreciate that. The Bishop, what's going on, Bishop? SARS-CoV or coronavirus, which is CoV, is sequenced. So this is another part of that same study. I have the link below for that study as well. It was reverse transcribed and transfected in Vero E6 cells, and this was 2003. And Vero E6 cells are the same cells they're using right now for coronavirus studies. And the Vero E6 cells are basic immunology cells that you use. I forget the actual tissue, but they're just basic immunology cells. I don't work with them, and I'm still trying to learn more about their morphology and function. But at any rate, they say here that the IC SARS-CoV marker mutations, cDNAs spanning different BGL1 junctions were amplified from wild type and IC SARS-CoV infected cells. So wild type are cells without infection, and IC SARS-CoV are infected cells. And BGL1 is a restriction enzyme that's used to cut different parts of the DNA. And we know that coronaviruses are RNA genome. So you use a restriction enzyme to cut that. You can put in a new sequence and then look and see where that sequence falls on using a gel. So let's come down here. I'm looking in their figure legend. Where is A? Oh, here it is. Okay, I see SARS-CoV coronavirus lanes 2, 3, 6, 7, 10, and 11. So 2, 3, 6, 7, 10, and 11. And the wild type SARS coronavirus lanes 4, 5, 8, 9, 12, and 13. So either way, there's differences in how this is being expressed, how the expression of these different cDNAs are being expressed. 2, 3, and 4 seem to have the same expression, and 2 and 3 being from the IC SARS-CoV infected ones. And then let's see, we have 5, 7, 10, I think, and 13 as well that are expressed much differently. Then you have wild type in B. You have wild type SARS lane 4, 5, 8, and 9 that are expressed from wild type. And then you have 2, 3, 6, and 7. 2, 3, 6, 7 that look totally different. 4, 5, 8, and 9 have one band. 2, 3, 7, 8. Okay, well, 2 and 8 have one band. 8 is the same as 9, same size. 2 is the same size as 4 and 5. But 3 and 7, you got two bands here. That means you have not cut that. Because if you have a big piece of something and if you cut it one time, you have two pieces, right? So that means it's much bigger than that. You have two bands there. I think this is 0.5. So this is 500 base pairs. I'm thinking that's 500 base pairs. Yeah, because it says 1 KB ladder. So it's about 500 for this one and maybe 200. So that's 700. and this is about 480, maybe 490, and this is 1 point, maybe 1.3, 1.4. So you have to add those together to get the total sequence, the total size that you're looking at. But at any rate, they see differences between the cells that are infected and cells that are not infected. Let's see. Um, what's going on, Seven Coast Dojo? Um, oh, and that word that y'all say, the OSU, the O, that's in the Daughters of the Trade. And I was like, why do they keep saying the word for the Daughters of the Trade? But it's a city, and I was like, okay, but hello to you. I just thought that was something interesting. Um, but I understand that it is also, I think, a greeting. So hello to you. Nameless said respect to you as well, Nicole. And thank you for sharing this research with us. Oh, no problem. No, the pleasure is mine. Thank you, Ronan. Ronan, hey. He said Linus mental powers activated. Thank you, sir. I appreciate it. My volume dropped. Okay, I might have to. Y'all know what? Let me go ahead and let me stop sharing real quick and go back to some settings I have here. Mm, okay, if y'all can hear me, let me know. Oh, let me see. I might have to change the display because they were doing something outside earlier.
I was trying not to have that in the background because it's really annoying. I don't know what they're doing out there. Anyway, uh, my bad for that, y'all. Let me go back to this sharing the screen. Okay. Mm, I'm not worried about what, they got a bunch of stuff going on back there. I ain't worried about that. Yes. Yeah, whatever. But anyway, let me come back. Doug dropping off for the offering plate. I'll listen later. Oh, no problem, Doug. Thank you very much for that. I appreciate it. Seem to come back. Okay, my bad. Y'all gotta tell me if the volume is all jacked up. Y'all gotta let me know. You're like DMX up in this joint. Y'all let me know. Okay, my bad for that. Because I had the volume turned up as high as I could. Everybody's putting one in the chat. But I'm like, I feel like DMX, y'all. You gotta let me know. Okay. Let me go back to this. Okay, so if y'all can see this, put a one. Thank you very much. SARS coronavirus sequence have been, I should say sequences have been reverse transcribed and transfected in viral E6 cells. So this is the reverse transcription that I was talking about earlier. What's going on, Tiger, MLR, Vernon King? How you doing? Y'all keep hitting that like button. Thank you so much. So this is where you see some of the manipulation that was occurring. This article was published 2003 as well. This is the same article I was talking about before. And all it's showing is that there are differences in the open reading frame expression, the levels of expression, these sequences. So you see these sequence changes that tend to occur right here. This is the mRNA species. These are the transcripts. This is the number they give, the code they give, the transcript. They have the leader sequence that you see here, which is identical for all of the different open reading frames. And here's where the sequence changes tend to occur. And you see that where you have infection rates that are varied based on these changes in sequences. That's all this is really saying. Moving on to from the mid 2000s to 2008, we saw a lot of articles being published. Of course, this is coming into the pandemic of coronavirus and the first, SARS coronavirus that we saw. And we also see that there are, what I have highlighted here, several studies where they have recurrent mutations that are associated with the isolation and passage of SARS coronavirus. Let me see something. There we go. Okay, so it's good. With the isolation and passage of SARS coronavirus in cells from non-human primates. So this is where they are going from one model to another, model jumping. They're going from models that are small cells, in vitro analysis, into in vivo analysis, which means in a living organism. We also saw that the SARS, which is severe acute respiratory syndrome, coronavirus infectious cDNA clones, and a replicon to study the coronavirus RNA synthesis. So this was important for me because this is kind of what I do, developing cDNA clones and cDNA clone libraries and different replicons that you can use to study a disease or something. And another study was they have a mouse-adapted SARS coronavirus, which caused disease and mortality in BOB-C mice. So these are little white mice that are immunocompromised, modeling a person who's infected with SARS. Another one where they have synthetic recombinant bat SARS-like coronavirus is infectious in cultured cells and in mice. So they took those cells that were infected with the SARS-like coronavirus and they infected the cells with it, took those cells and put them into the mice, and they were able to transfer infected cells into mice. So the mice then have the disease. Okay. Then coming down, all of these 2005, follow dates. I always follow dates. 2005, 2006, 2007, 2008, another one published in 2008. Animal models and vaccines for SARS coronavirus infection. This is from Virus Research publication. This was also 2008. So you see that the development of very sophisticated technologies and studies on coronavirus is not new. It's been going on for a long time. Now, I pulled out one study, which was the last one here with Roberts et al. From 2008, and I looked at some of the data that they have here. One is the histopathologic features of infection of SARS coronavirus in the lungs of aged mice. So they have 3 different strains of mice. They have the 12956, B6, and BOB-C mice at 3 days post-infection. So they're infected with coronavirus. 3 days later, they sacrificed those mice, did immunohistochemistry on the mice to determine if there was expression of this particular protein. Let's see where. So the proteins you're looking for are antigens against SARS coronavirus, and that is what you see in red. So these red areas, this is inflammation with coronavirus, coupled with coronavirus. Anytime you see anything black or I wouldn't say gray, but anything that's really dark black or just really dark brown where you shouldn't see dark brown, like this is inflammation of the lungs. This area is probably not as inflamed, but it may be becoming necrotic. This area is on its way to being inflamed due to this area here that's extremely inflamed. And it's also indicating with the darker red color that it is coronavirus antigens that you're seeing here. So how do they know, or how do they create an antibody against a virus and, against its antigens? Well, we already know, right? We know the sequences. We know the antibodies, the protein sequences. So you can develop an antibody when you know these sequences, because you can develop a synthetic antibody from this protein sequence. So that's how it was done. In my humble opinion, that's how it could have been done, right? And so we come here in B, and B is showing that there's abundant SARS coronavirus in antigen being expressed here in the cytoplasm of these cells. It's red, and anything that's like a lighter gray color is not the expression of that antigen. In C, there's necrosis occurring and inflammation here. There's a lot of necrosis here in the middle of this tissue. This is respiratory epithelium and inflammatory cells. So not only do you see inflammation of the epithelial cells, but you see inflammation of the inflammasome pathway. The inflammatory cells that should be helping you, they're wasted. They're done. They're like, look, I can't do no more. I can't fight. Which is why they do the infection in mice, the murine model or mouse models or murine models they use. They'll do it for 3 days, no more than 7 days, because 3 days for a mouse is like a week and change for a human. Okay. We come to D, we have more IHC or immunohistochemical staining of the same section in C. They have it in D. And this time it was for the antigen in a different cell, and it was looking at intraluminal debris. Intraluminal debris is the debris that you find within the lumen or open space of tissues or organs. Here they focus more on the interstitial inflammatory cell infiltrate from a specific strain of mice, which is biopsy mice again. And in F, they give the SARS coronavirus antigen in alveolar pneumocytes that are adjacent to the bronchiole in the biopsy mouse again. And then they have, the H&E staining that they always show it as a counter stain that you have here. And the counter stain, which is that some of this not really, a little bit like pinkish color in the background. Some of that indicates inflammation. It also indicates histology that is not healthy. The only problem I have with this image is they didn't show a healthy panel next to it, but I believe it was in their supplemental data as well. Remember, this study was done in 2008. This is another study done in 2008 as well with this similar idea, where they say, let me hide that. This is from Becker et al., 2008. So I have Becker here. Synthetic recombinant bat SARS-like coronavirus is infectious in cultured cells in mice. So Sway, how is this happening? Okay, well, I'll let you know how it's happening. So you have a schematic of SARS coronavirus, open reading frames, RNA binding domains, things like that, different mutations and cuts and inserts that have happened through evolution. SARS coronavirus here. You have the SARS coronavirus, which would be the human, bat SARS coronavirus, and you see differences in the RNA binding domain here at E2. You have bat SARS coronavirus, a different type, where you have differences in the expression of that. B is another indication that there are differences and there are a lot of mutations that are occurring. So you have your standard consensus sequence here, and you don't see the same thing when you look at the bat SARS coronavirus. And then they looked at it by mRNA on a gel, and you don't see, if it was all the same, you just see all the bands going straight across, but you don't. You have gaps in the expression, and you have different levels of expression. So this band here is darker than this band, but this band is lighter than this band. It's not as heavy. So the expression levels are different. I'm gonna stop and come back to the chat. Okay, Ron Reed, how are you? Did appreciate what you're doing. Oh, thank you, Ron. Yes, I got your email. I hope that the email I sent you was helpful in what you were asking me about. If not, you can definitely hit that link and come up here. Nameless said, can you explain a cleaved protein? Is that a sign of editing potentially? No, cleaved proteins are cleaved all the time. You have enzymes that cleave proteins. And if you did not have enzymes cleaving certain proteins, then we probably wouldn't be here, because those are required to cleave a protein and prepare it for its function, its final function. Like if it's not, every protein doesn't need to be cleaved necessarily, but a lot of them are cleaved to do something, okay, to whatever function it is. Maybe it's a protein that needs to be cleaved so that it can bind properly to a lipid or fat. Maybe it's a protein that needs to be cleaved because it needs to bind to a carbohydrate. Maybe it's a protein that doesn't need to be cleaved at that time, but due to a disease, it's cleaved too much. So that could contribute to a deficiency or a disease. And so it doesn't necessarily mean that it's a sign of, artificial editing from somebody like myself. What's going on, Shango? He says, salute sis for this much needed info. Oh, no problem. Thank you for coming. Thank you. Yeah, get them likes up, y'all. Get them likes up. AN1 said, but did they put only live culture viruses into those mice? More than likely they did, because I put live viruses into my mice because they're mice, and you just want to see the most potent effect. We do attenuated viruses as well, but we'll put the virus into the cells, and then put the cells into the mouse. So if the cell has active virus sequence in there, then yes, that is indirectly like putting a virus in your body. It's an indirect type of thing. Some of the studies, like the one that you and I talked about from 2020, where they looked at SARS-2 in mice, that was a artificial sequence that they used to put into the mice, but it's the sequence that's exactly the COVID-19 sequence. Yes, it is a copy paste behavior. In some cases, nameless. No, I do not, India. She says, do you trust the release of a vaccine within this year? No. The nameless protagonist. Okay, so it's not a sign in and of itself, but it can cause problems like a deformity of cleaving needed to occur but didn't. Okay, yeah, no problem, Keith. Wisdom, no problem, Keith. You're welcome. So virus is conducted from the mid 2009 to or mid 2000s to 2014. Actually, there were so many to put in here from the mid 2000s. I just left them out because you wouldn't be able to see the words on the screen. But this is just an example. I've only highlighted 4 on the screen, and these are studies conducted from 2015 to 2016. Of course, there are many, many more I have not added here for brevity. I've highlighted 4 that were interesting to me. And like I said earlier in the stream, that you all, if you want to have a copy of this PowerPoint, I can send it to you. You have to email me and let me know. Thank you, Ron Reed. Thank you, MLR, for the Cash Apps. I appreciate that. Okay, and here is another example of comparing the Middle Eastern respiratory syndrome to SARS, and this study was conducted in 2016. So there were 2 coronavirus type issues going on. You had MERS coronavirus and you had SARS coronavirus. Once again, these are not COVID-19. These are before COVID-19. COVID-19 is based on novel coronavirus 2019. Okay, so what they show here is analysis of the MERS coronavirus on the top and SARS coronavirus on the bottom, where they use the CALU3 cells, I'm reading here at the bottom, based on differential gene expression profiles. And what they find, if you just have a cutoff point, you have to have some kind of cutoff point. And they chose here that this will be their cutoff point. The MERS coronavirus seemed to be more infective than the SARS coronavirus. And so the idea from this data is that SARS coronavirus, even though it wasn't as infective or effective as a virus in patients, the mutation rate was higher in SARS compared to MERS. So why do I say that? I say that based on several pieces of data from this paper. This is only one, but the expression of cytokeratin 18, CD31, which is surface protein, and synaptophysin, which is a neuroendocrine marker or hormone-dependent marker that would regulate a lot of the changes you see in these functions, the cells that would function, do these functions, especially the kidney, the liver, and the heart. Those are the 3 main things that they're looking at in this study. So a lot of the expression level should be high in the MERS COVID-19 virus, which is in green, and these proteins, which are in red, it should be high, and it is high. You see that they have a protein against MERS coronavirus. It's in green, and cytokeratin 18 is in red. So wherever you see green and red together, is going to be right here. So these cells do express cytokeratin 18, which is a functional protein that goes up when you have this type of infection. CD31 goes up when you have this type of infection as well. So they have MERS here, but you don't see CD31 here as well. It should go up in this type of infection, but it didn't. And so when you merge these two pictures together, that's what this is. This is a merged image. You only see green. You don't see the overlay of the red and green together. And synaptophysin being a neuroendocrine marker, you have that here. With the MERS expressed, you have less expression of a neuroendocrine marker, and you don't have much overlay. You don't have much yellow, because when you put green and red together, fluorescence, you usually get this yellowish or orange color. You don't really see that, expressed, especially in this portion of the cells that they're looking at. You don't really see that there. So it's indicative that there are differences in the expression rate of these markers for a transitional phase of these cells, at least with MERS infection. And you also see differences in expression of different genes when at different hours. When you compare MERS with SARS, you only have expression here in lane 6, and that's it. You don't really have expression level increasing as you do in MERS. So although, once again, all MERS coronavirus is more effective compared to SARS coronavirus, possibly the mutation is more important to look at with SARS. And why do I say that again? There's more research to show here. Let's kind of ignore this part. Let's move on here. This is just indicating that this is indicating that the MERS is more effective. It kills. This says relative apoptotic activity, meaning death. Apoptosis is cell death. MERS infected cells is way up. This is significant compared to control, no infection, and SARS. MERS is extremely high. Okay, the same is true for SMAD7, FGF2, and FGF2 production. So there's a difference here, but I'm going to go and show this mRNA expression of SMAD7 way high in the MERS infected cells. FGF2 is fibroblast growth factor 2, way high in the cells infected with MERS compared to SARS. And this is FGF2 production. So FGF2 is a factor. It is a ligand that the cells produce when they're stressed during immune assault or an infection. So they have captured FGF2. Not only did they look at the mRNA of FGF2, but they did something different. They also looked at FGF production. How much of this ligand, how much of this protein is expressed? And it's much higher in MERS than SARS. So when people say that SARS wasn't that bad, they lying to you. Roger Report. Oh, hey, how you doing, Roger Report Live? Thank you for that super chat. I appreciate that. BGS, what's going on, Master Teacher? How are you doing? Oh, wow, everybody's in the chat. Thank you so much for coming. So when people say, I thank y'all, appreciate y'all for coming, because I was like, let me try to do this real quick. But when people say like, oh, SARS is not that bad, they're talking about SARS, the first SARS. Okay. I'm gonna say that again. When they're talking about SARS ain't that bad, it's like a regular flu. It ain't no big deal. I don't know why everybody here wearing these masks and shit, doing all this move. Like, sit your dumb behind down. Okay, you talking about 2016. You talking about 2005, 2008. You ain't talking about like right now. Please sit down. Anyway, that's just, it kills me when people say that. Why are you talking about this now? Okay, sit your slow behind down. You want to read something or just be quiet. So when we look at like B, I know I'm jumping all over, but let me show you B. This is protein expression. Okay, how do you know? It says Western blot analysis of caspase-3 SMAD7, which is a factor in transitioning cells from less aggressive, like a regular epithelial cell, to an epithelial cell like a cancer cell, or a less normal cell, a cell that's more likely to be correlated with disease. So SMAD7, I deal with SMAD7, SMAD4, SMAD5, SMAD2, 3. Yeah, SMADs are not good. When they're up, you have a problem. You have a disease going on. Caspase-3 is expressed when you have a lot of death in that environment, that cell environment. FGF2 is usually expressed in fibroblasts, but it's also expressed in immune cells. So that's important too. FGF2, FGF7, FGF10. So they just chose one of the genes from each group. Caspase-3, there's others. There's caspase-6, caspase-8, 9, 1, 3/7. You know, they couple each other. So they just chose one gene from each of the types of groups. You know, genes are classified by their function. And you can do all of them if you want to, but you're not going to be happy because they're all going to be different. So that's why they chose just 3 to make it simple. Either way you slice it, MERS, which is M, compared to control, which is no infection, versus SARS, which is infection, you have way high, these cells are dying, cleaved caspase-3. This is a cleaved caspase-3 expression, similar to what we just said earlier when I was talking to Nameless in the chat about a cleaved protein. Cleaved caspase-3 is active caspase-3. Total caspase-3 is not cleaved yet. So it won't be this big. The expression level will be very low. Okay, once it's cleaved, you have a lot of it. It's active. SMAD7 is much higher. It's not detected. They're not detected. FGF2 is here, very high. It's not detected. MERS, COVID, NP. I think this is what was NP? I forget. NP was I think normalized protein for MERS and SARS. So it's expressed here, but of course, the SARS too, it doesn't have it with MERS and it was with SARS. So this is your positive control. So you have to have a positive control when you deal with these viruses. Apparently, this is a normalized protein only for MERS. It only shows up in cells infected with MERS. It does not show up in the other 2 groups. The ones for SARS, normalized protein, does not show up for MERS or control. It only shows up for SARS. And gamma tubulin, gamma, oh gosh. Okay, they use gamma tubulin. All right, I ain't never heard of it, but that's what they use. Okay, gamma tubulin. So there's 3 types of tubulin. I just realized what they use, alpha, beta, gamma. So I use beta tubulin. They use gamma tubulin, which is something that is specific for certain cells apparently. And this is just a loading control to make sure that whoever did this experiment, they loaded the samples properly. And you see the expression is the same all the way across. You should have expression because you're using the same cells. They're just treated differently, different infection. This is a heat map that indicates that there are 2 genes that they want to focus on, SMAD7 here and FGF2, which had high expression in the MERS group, which is on the left side, versus the SARS group on the right side. So you see FGF2 is high expression, which is red. And it's almost non-existent. It's almost at 0 for the SARS infection. SMAD7, there's not much difference. I mean, it may be significant, but it's still somewhat red. But they wanted to focus on SMAD7. And then you look at this other set of genes, and it's low for both. You know, it's closer to -3, maybe -2 for most of these genes for both groups of infected cells. So, for that study, the focus was they were trying to determine if SMAD7 and FGF2 could be markers for MERS infected patients and SARS infected patients. And it seems like that's true. So there are more studies from 2017, 2018, 2019, and 2020, and 2020, and one more 2020. So they have a lot going on, as you can see. And all I've done is compiled what I could find on these particular studies. I use the study that I'll talk about in a minute to like wrap up what they're doing right now and how things have progressed since the early 2000s and even late 1990s. Okay, so I just go to the chat real quick while I put that up because, you know, let's see. Yeah, it is interesting how they do it because and these genes, nameless, are compiled from a lot of the um, genome databases, a lot of the things that I show on here on my channel, NCBI, PubChem, a lot of the different websites that just give you a lot of information about the function of these genes. And some of the genes we don't even know, like some of the mutations of these genes, we don't know exactly what they do, but we have an idea. They can tell you based on sequence homology and um, different organisms what they think it may be doing. And so it'll say a putative gene function or putative gene sequence. And so that's helpful as well. But I hope that this is not going too far. I know I did this last time. Yeah, she's literally saying, I'm overwhelmed. I can't. So yeah, I'm going to pause there and oops, come out of here. Let me stop there. Okay, let me see. Okay. So I hope that you guys are learning something. I'm stopping there because I feel like if anybody's, lost or, anything like that, or they want me to go over something else, because I have about, I think I only have about 20 slides left. Yep, I think I only have about 20 slides, but I'll just stop here for a moment. VGS said we're going to have to watch this like 3 times. I'm sorry. I'm trying to make it as simple as I can. I have to put PowerPoints together for a lot of my work anyway. And so, I really am trying to make it as simple as I can to break it down to bits and give it to you. I hope I'm not going too fast. I was like, how am I going to do this? Nameless said it's good. They identify the sequences ASAP and they have a library of info to make these correlations. Again, the immaculate conception theory for COVID is not exactly, that is not holding water. I didn't believe that when I first heard it. I was like, wait, we already had SARS. Like, you have to have a baseline sequence to go from in order to develop something like this. And I was thinking, I thought, no, I didn't think it was immaculate conception type of idea. That theory is in the trash for me. But I did think that it was perhaps a mutated version of the original SARS. I was like, oh, maybe it's mutated so bad that it has become very virulent. And the virulence rate is, the R0 numbers, everything is so high. Maybe it's mutated, but I was like, And then I started to think, well, when has a virus like that mutated that quickly? Without anyone's notice or anyone trying to interfere with that. That was weird. So it didn't make sense to me. What's going on, Sachi? What's going on with you? This was a masterclass for this topic. I'm just trying to make it more understandable. Nameless said also giving us good lexicon to work with, like cleave proteins, differences in RNA and DNA, as well as how control groups can be marked to isolate sequences, alpha, beta, gamma. Yeah, they do. We have differences in how we analyze the data, and also wanted people to recognize that there are differences, or the advancements in how the technology is being used. They've developed new technology based on what we're seeing with these diseases. Double C, what's going on? 20 slides, that's it. No, I have 41, but I stopped at slide 20. If you will have to digest it. I hope that people digest it and they can glean something from it. It will help them. And I agree with Nameless, because it helps people when they go in real life and they're talking about this topic and other things. And they can say, well, that's not how this was done. Like, this is how it was done. And I can, they can show links and, documentation to prove what they're saying. Oh, no problem, Sachi. Finish the slides for our archives. I mean, I was waiting if anybody wanted to come up and say something, but if not, I'll just go on. Let's go on, Micah. Yeah, all is well here. Everything's good. I wasn't able to continue. Like, I'm not able to, of course, if I'm doing all this stuff, I'm not able to like highlight your comments and stuff. I only have two hands, I haven't been able to build. I haven't been able to build another set of arms for myself. But anyway, moving on. Moving on. So this is okay. So this is a SARS COVID-19 sequence that they show is totally different from previous coronaviruses, and it is showing a reference genome sequence. So severe acute respiratory syndrome coronavirus 2 reference genome. The open reading frame AB, or 1AB, excuse me, and the open reading frame 1A are at different sequences. There's a huge gap here indicating some mutation that has occurred. And there's gaps here. There's all types of gaps. So we went through this a little bit on Dr. Dunbar's stream last Saturday. But the reason that I wanted to show, so why am I showing this? I'm showing this because, one, is easily accessible. All you have to do is type in COVID-19. The first thing you type in on NCBI is COVID-19. Let me see here. I went to ncbi.nlm, a National Library of Medicine, National Institutes of Health.gov, and you go to the search bar and you just put in COVID-19. That's it. And you'll get this. This will be the first thing you will see is this sequence. It's a single-stranded RNA virus, period. That's what they have. They've gotten this sequence from a host, which is a human. So I went through this study before. I wanted to go through it a little bit more again. This was how the SARS-CoV-2 infection model in mice was demonstrating protection by neutralizing antibodies. So neutralizing antibodies were produced after infecting these mice with human adenovirus, human ACE2, which they don't naturally have. They're mice. They don't have human sequence for ACE2, the angiotensin receptor. So they had to put that in them and transfect the mice with that. Then they gave them the nasal injection or transfection. They gave them SARS-CoV-2, which we're dealing with now. They have them give them ACE2, 5 days later, give them SARS-2. They get the infection in the lungs and throughout the body, and it helps them understand the disease, test therapeutics and vaccines. But in the process of them being infected, they develop these antibodies, monoclonal antibodies in their blood. You can take that out of them, take their blood, put it into another mouse, and you will have protection against coronavirus. So it will block what they have here, will block these effects. That's the study in a nutshell. They have highlights here and everything. I'm going to move forward. I've already shown this picture. This picture I've shown, but it's very important because it's indicating the different organs that are infected with coronavirus. This is the log base 10 number of copies per milligram or microliter. So per milligram of tissue or per microliter of blood. Okay. SARS-CoV-2 viral RNA. 4 days post-infection. DPI means days post-infection. They gave the mouse the infection, and then 4 days later, they killed them and took out their organs and looked at them. So as you see, they have them in order pretty much almost from most number of viruses to lowest number of viruses or viral copies. So the lung has the highest, and then the heart, And then I think Dr. Dunbar mentioned this on my last time I talked about this study. The spleen was actually third. And for me, the liver is fourth. The brain, it's not consistent. The pattern of expression of these viral copies is not consistent. There's no pattern. It's all over the place. This could be due to the age of the mice. It also could be due to their immune systems and the efficacy of the blood-brain barrier of the mice. That's what this could be due to. But you have the lung, the heart, the spleen, the liver. Now these two make sense. I thought it would be the other way. I thought the liver would be higher than the spleen, but these make sense. The liver and spleen are, physiologically right next to each other. So that kind of makes sense. Usually what happens to the liver happens to the spleen as well. At any rate, you see that you don't have any significant difference in the low or high copy numbers of these particular viruses for the lungs. There was no difference. It was high. It was very high for all of them. 4 days post-infection, very similar to the lung that they have here for these different viruses. The different colored dots are the different viruses with mutations and whatnot. Okay. So then another thing that was interesting to me was they looked at lung titers or lung viral load in BOB-C white mice immunocompromised versus lung titers of C57BL6J, which are black 6 mice. So C57BL6J are not as immunocompromised as BOB-C white mice. We call these black 6 mice. They are black. And usually they can survive longer than other mice with certain infections and tumors. So a lot of times I don't even use black 6. I like X because they're not gonna die fast enough. And what you want to do is limit the amount of pain that the mice are going through, because that's unethical to let them just stay in pain like that. So since it takes too long and they get expensive, I'm like X. But they use them here because they saw that there was no difference significantly between 1 day post-infection, 2 days post-infection, 4 days post-infection for both mice. They both had the same number of copies of virus, around 7.5, is the average here. So that was interesting to me. Another thing that they showed, they could recapitulate this infection to analyze organ differentiation and viral pathogenicity. And what they did was they extended the time after infection before killing the mice. So 8 days post infection versus 4 days post infection and 0 days post infection. So this is no treatment. This is normal. Normal, no treatment. Then you have adenovirus. You have adenovirus SARS-CoV-2, and then you have this alpha IFNAR1 SARS-CoV-2. This is a mutated COVID-2. This is a standard COVID-2. So this is what you want to avoid on the street. This one. So 4 days, 8 days. And we just look at the histopathology at 4 days versus 8 days. You can't really tell much of a difference at this. So they made it bigger. They increased the magnification. So we look here and we see there are changes in the morphology, in the structure of the mice lungs. And we see here that there is a huge difference. There's more red. There's more necrosis perhaps that's happening. More red, meaning more tissue that's available for inflammation and necrosis. And then we look even closer with the magnification, at a higher magnification, you see that there is more damage that's occurring. In this 4-day picture of SARS-CoV-2, you see that there's not much infiltration. There's a little infiltration here, these small dots, some of these small dots here. There's infiltration in the lumen of the tissue that's breaking down. Here you have lots of infiltration that's occurring. So infiltration in the immune community, a virology community, is indication of breaking away or destruction of tissues. It's also indication that your viral load is going up. So Let's see. Oh, okay, no problem, MLR. I had to check my email. Shop Talk. She was going on Shop Talk. He said, sorry, I'm late. My dog ate my test tubes. Man, I can't use that excuse. If I could, man, you know, I've been able to get out of stuff, but I cannot. Mike V, what's going on? How are you doing? He said, peace to the chat. Peace to you, Mike V. So when I'm reading this stuff, this is how I feel sometimes. I'm like, don't bring me no bad news. I don't want to hear it. If anybody, you know, you saw the movie The Wiz, then you know that what the song is and what this quote is pertaining to. Don't you bring me no bad news, no bad news, no bad news. When I started reading about this is how I feel. I don't want to hear no bad news because y'all don't understand. I was dealing with a study that wasn't working, and we had so many different inconsistencies of our data. And I was like, oh my gosh. Then we have this come. We didn't really think initially that it would be this bad, but it is this bad. So I have to retract my statement on don't bring me no bad. Exactly, AB. Don't bring me no bad news. That's how a lot of people feel, I think, right now as well. Is they're just like, I don't want to hear about this mess. You know, it's just like a regular flu, blah, blah, blah. I'm tired of that. It's not. It's eviling. Yeah. And so I feel like, you know, you have to go into it not with your, you know, head under the bed. You know, and I myself have to admit, you know, I can't just be like, well, I don't want to hear about it. It's just bad news. I got too many things to do. No, you should try to understand a little bit of it. You know, just try to be a little bit more aware of what's going on. You know, and so I have here, you know, this is good news, in my opinion, for the study. Once again, study conducted 2020. And they have recapitulated the mice to with the SARS COVID-19, aka SARS-2. So they're infected with that. They're infected with this human ACE2 adenovirus. They're given SARS-2. They're given this 1B07 or isotype by IP injection, meaning in the interperitoneal injection at day 1. Then you're counting backwards. Then you give them injection of SARS, intranasal administration of SARS-CoV-2, and then you see if it's gonna reverse the effects of SARS-CoV-2 infection. Okay, so this is how they've set up their study for this part of the project. And what we find here, so you have the isotype, which is 2FO5, or 1B07, which is the neutralizing antibody. And what happened with the percent relative infection, it went from 100% at day 0, and in 4 days, it's dropped down to almost 0% infection. The weight change actually went up. So when mice lose weight, they're dying. And the isotype here, they're dying. I mean, their weight change is just dropping. And I don't think that these two would be significant. I don't think these two groups would be significant if they didn't increase the concentration of the neutralizing antibody, because they did in their methods increase it. And so that's where you see this nice increase in weight, weight gain, where they're increasing in weight. And that's significant, indicated by 3 stars or 4 stars. So they're comparing the blue dot here to the black dot here, the blue dot here to the black dot here, and the blue dot here to the black dot here for day 2, 3, and 4. And I want you to recognize that this induction does not occur until 48 hours after infection. Okay, the lung titers at 4 days post-infection. Black is the isotype or control, where you don't have any help. And the blue is where you have help with the neutralizing antibody. It dropped. So the amount of virus drops and is consistent within that group. Lung viral RNA is different than titer. Lung viral RNA is where they had to do RNA isolation of the tissues of the lung, and specifically look for RNA that is not the mouse RNA, and RNA that is not human RNA. Because remember, these mice have human ACE2 in them as well. So they had to do this for the virus sequence, RNA sequence. And what they found that is a decrease significantly of the number of copies of RNA per milligram of lung tissue. Okay, the same is true for the heart. However, the heart data compared to the lung data is not as, the spread is different. And this was problematic for me because this data in the lung is more statistically significant. They have 4 stars. The heart viral data that they have, the RNA, is 1 star. So it's 0.05 p-value. This is 0.0001 p-value. Okay. So I have a problem with this data, but that's just me. The spleen data is fine. It's very similar to the lung data. Actually, it's decreased more than the lung data by about 50%. The control is about 50% lower than the control here in the lung. And it drops down, to from about three to about 2 or so, average about 2 copies per milligram of splenic tissues. But this one is still problematic to me. And I hope that in their discussion, I have to go back and look, that they discuss this because people were having heart attacks, strokes, all kinds of issues going on that are cardiac involved. And so that I hope that they have better data at some point for that. Then they go into the specific gene expression that occurs and that changes when you give mice, this neutralizing antibody, IBO7. So the lung, they have IL-6 or interleukin-6, which is an immune system regulator. You have CXCL10, CCL2, CCL5, CXCL11. You also have interferon beta, IL-28 alpha beta, interferon gamma, tumor necrosis factor, and IL-1 beta. So when they put B, it means beta. When they put an A, it means alpha, usually. But this is how you would write a gene in lowercase letters. But so they have a decrease. All these genes, they're decreased in expression. Here, interferon gamma, no change. Tumor necrosis factor, no change. IL-1 beta, no change. Why? I don't know. I could not figure out for the life of me when I'm looking through some of the data why interferon gamma tumor necrosis, I think this is TNF alpha they're using, and the lung IL-1 beta, why there's no change. But at any rate, they did see significant changes for a lot of these markers. And these are markers that you will look at for a clinical panel for patients. If you were looking at patients infected with SARS to COVID-19, this is the type of panel that you would use after treatment. Okay, so they say that this neutralizing antibody, monoclonal antibody that they have developed in their lab called IBO7 has a protective effect against COVID-2 infection. So that's very good. And they also say they gave this, mice were treated via intraperitoneal route, 200 microgram of IBO7, or to control, which is influenza A virus, monoclonal antibody, and then after a day, they inoculated them with it in this route. So yeah, intraperitoneal is just like in the stomach, basically. They just did an injection in the stomach. Yes, cytokines, Dr. Dunbar. Yes, I think you know more about cytokines than I do because of the pharmacology and toxicology background that you have. I know of IL-6. I work with IL-6 a lot, and I work with interferon gamma a lot. And I was kind of bummed that, it didn't do what it's supposed to do, but these are mice. We have to remember that as well. So perhaps interferon gamma is not expressed at a high level as it is in humans. That could be one thing, but IL-6 is very, very critical to see that, and to see the CCL5 as well decrease. And I want to give a little bit of insight into CCL5. CCL5 is a chemo-like, I think it's cytokine chemo-like number 5. So you have CCL5, which is proponent in a lot of HIV studies, and you have like CCR5 and you have CCL5 Delta 32 mutation, which occurs in people who are protected from HIV. So the CCL5 Delta 32 mutation was something that the doctor in China, I think it was last year, tried to do with the young girls that were twins. So using that in viral studies is a standard. It was a standard before he tried to manipulate CCL5 in those girls, which we don't even have data for, but he said that's what he did. So that's a standard. CCL5, CXCL11, CXCL10. Actually, another one is CXCL4, IL6, IL12, IL10, IL8. These are standard genes. So it's not like they just pull these genes out their butt. Please don't think that's what they did. And this study, this image I showed before from the study where they have the control here, very high levels of infection after 4 days after infection. And then you add neutralizing antibody, you don't see any more of the red. It really not that much. I mean, you look at these two images, it's like night and day. You know, you see these two images, let's blow it up. You know, you see these two images. You have on the left side, you see that it is very red, very infected, lots of virus. And on the right side is really nothing there. It's almost completely gone. And we have to remember that this is 4 days after infection. This is not, 10 days or 14 days. We have to remember that. So, they're just showing the pathology of the infection and what's happening. And the same is true here when you look at the H&E staining, the hematoxylin eosin staining. You have the control and it's reduced, is somewhat reduced. For me, at 6 days post-infection, I would expect it to be much more reduced than this, but that's what they have. This is the data they have. So let me read the chat real quick before I transition again. Roger Report Live said, I'm just here to pretend that I understand science the way you are. You don't have to pretend. I don't know everything. I'm trying to learn, man. He said, it's never too late to learn. I'm just here pretending I understand this stuff, man. No, trust me, like it's stuff that I don't know, like that Dr. Dunbar knows, like, cause I'm trying my best to understand. Immunology is not my strong point. I do not like immunology in some cases, but I'm having to learn on the fly. So join the club. You're right there with me. I'm still learning this. Nameless said these conversations happen in the scientific community. Yes, they do happen. I just had them earlier today in my meeting. He said, I wish they talked about stuff like this in school. Far more of us would be in STEM. Yes, absolutely. It will sink in. Just 10% is more than a lot of doctors know. So I guess that's probably true. We just I hadn't really thought of it, but a lot of physicians don't really they don't really read the research, and we have to tell them, hey, we just did a study on that like 2 weeks ago. We did a study on that last year. And they're like, what? Why didn't I know about it? I don't know. Aren't you the doctor? We're looking at them like, aren't you a physician? Don't you read? And they were like, well, we read, but we don't read everything. I'm like, oh, okay. All right. Thank you, Go Professor. He says, supporting the channel. I'm with you, Nicole. Salute and salute to the chat. Thank you so much, Go Professor. Nameless antibody treatment is good news. Yes, I think the antibody treatment and the mRNA vaccine that they're developing, I think those are good options. He said the problem is infection doesn't necessarily correlate to external symptoms. So folks won't even know they need to get treatment in that 4-day window. Well, they did the 4-day window to mirror like a 14 to 21 day window. I'm not 14, like 7 to 14 day window. He said the antibody treatment, like it is good news, but the problem is the infection doesn't, so it doesn't necessarily correlate to external symptoms. But the thing is, with a lot of these studies, what they'll do is they will document the physical features, the physical symptoms, external symptoms that you would see in the mice. And that sometimes does mirror what humans see. It's very similar to what humans have as far as symptoms. And I'm sure that there are some mice that had no symptoms. I'm pretty sure based on this, what we're looking at, I'm sure there were some that had no symptoms. However, those mice were not counted in the statistics. because you want to look at patients with symptoms when you're trying to use this type of antibody. Yes. So I have another section. This is, I think it's going to be the last section. Unusual features of SARS-CoV-2 genome suggesting sophisticated laboratory modification rather than natural evolution, and delineation of its probable synthetic route. All that means is we think SARS-CoV-2 was made in the lab. They should have just said that, but you know, they have to be extra scientific. I gotta tell you everything I'm doing. This they use all these, and what am I saying? Y'all probably think that about me. They using all these big words, and this is they put the word sophisticated in the title, delineation in the title. They were like, yes, give them all the big words so they can't figure out nothing except we're talking about SARS-CoV-2. That's all they gonna know. So this lady, Dr. Lee Min Young, she actually is the main person that's pushing forward with putting this information out there. Her correspondence is right here below, team.lmyan@gmail.com. They really went in this paper and broke down the reason why they think COVID-19 is, hypothetically, why it might not be a natural virus. It may not have evolved naturally. Okay, so I went through this before, but I know a lot of people weren't there at Dr. Dunbar's stream on Saturday. So they say here that there are 3 reasons why they think that there is in vitro or artificial or cell culture manipulation. There are 3 lines of evidence to support their hypothesis, and they say that the genome sequence of SARS-CoV-2 is suspiciously similar to that of a bat coronavirus discovered by military laboratories in the 3rd Military Medical University and the Research Institute for Medicine of Nanjing Command. 2 different labs, okay, that have this similar sequence. The receptor binding motif, or RBM, within the spike protein of SARS-CoV-2 that determines host specificity resembles that of SARS coronavirus from 2003 epidemic, and it's very suspicious. Genomic evidence suggests that the RBM has been genetically manipulated. So when they talk about genomic evidence, the genome is all the DNA of that organism, not gene number 1, gene number 2, gene number 3, gene number 269. No, genome means all of your DNA, all of it. Okay, y'all got me? So they're talking about a huge amount of manipulation that had to occur in order to get to what we have now as the sequence that we're looking at when it comes to SARS-2. I had to throw some country in there. I love it. SARS-CoV-2 contains a unique furin cleavage site in its spike protein, which is known to greatly enhance viral infectivity and cell tropism. So it enhances how the virus infects you. It also enhances what cells it's going to infect and where it's going to stay, where it's going to make you sick, where it's going to kill you. That is not normal. Yet, this cleavage site is completely absent in this particular class of coronaviruses found in nature. I just said that is not normal. So that cleavage site that you have in this virus is not in coronaviruses in nature, like the bat coronaviruses. In addition, rare codons. Codons are just the sequences of 3 base pairs at a time. You know, AT, G, TAT, AAT, GGC, things like that. Those rare codons that are not normal, they're associated with this additional sequence, suggests the strong possibility that this furin cleavage site is not the product of natural evolution and could have been inserted into the SARS-CoV-2 genome artificially by techniques other than simple serial passage or multi-strain recombination events inside co-infected tissue cultures, or animals. So that's telling you right there. That's telling you right there that it could be artificial. It could be something done by hand. And then they give you sequences. Open reading frame 8 is an accessory protein, or that open reading frame sequence on the genome actually translates into an accessory protein. The function is largely unknown in most coronaviruses, but they think that it could mediate the evasion of host adaptive immunity by downregulating major histocompatibility complex number 1, or MHC1. Major histocompatibility complex, what does that mean? What does that mean, Nicole? That is the complex, there's MHC1 and MHC2. That's the complex of different chemokines, cytokines, autocrine signaling, paracrine signaling that's required for you to have a standard host adaptive immunity or secondary adaptive immunity. All the T cells, B cells, the cytolytic T cells, cytotoxic T cells, the protector cells, your lymphatic system, all that type of stuff. So what they're saying is that this virus SARS-2 is genetically ready to hide from your immune system. It's genetically prepared to be like, well, I know that nameless protagonist, I know his immune system looking for viruses got on blue jacket. I'm about to wear red today. That's what it's basically saying. I'm about to wear red today because Gold Professor, his immune system looks for viruses in a green jacket. I'm about to wear yellow. Forget all that. I'm wearing yellow today. That's pretty much what it is. So, it kind of knows, like, most humans with their immune system looking for viruses that are wearing blue. I'm automatically going to wear red. Just going to go all out. I'm going to wear all red all day, blooded out all day. Nobody's going to see me. Nobody's looking for me. The surveillance, the police in your immune system, your innate immune system, macrophage, dendritic cells, natural killer cells. These are the police in your immune system. They're not going to see me. They're going to be like, oh, how you doing? All right. Carry on. They're not going to see me, but I'm the worst virus they've ever come in contact. I'm the worst criminal they ever come in contact with, but they don't even know it's me. That's what goes on. That's what's going on in your immune system. And on top of that, since I'm wearing red, I'm inside of Albert and I'm the virus, he's not going to have any symptoms. Albert walking around chilling, eat some Cheerios in a bowl. He's not even recognizing that he's infected. He doesn't even know what's going on. Not only does he not know, his immune system doesn't know, his innate immune system doesn't know, his adaptive immune system doesn't know, his liver doesn't know, his heart, nobody knows what's going on. That's the problem. That is not normal. That is not a standard virus. When you look at these sequences and you see the red is just standard. At the bottom, your consensus sequence, this is a standard sequence. I think I'm missing something. Somebody put, okay. What's going on, Q497? He said blooded out. Yeah, they're going to be blooded out. Like, I'm ready to go. So what's up? Like, what's going on? Like, these viruses, this is not regular virus, but continuing on, consensus sequence they have here is your standard sequence, and everything else is supposed to match as closely to this as possible. However, SARS, we have different SARS represented, and anytime you see anything that's black or blue, usually the blue is indicating a mutation. So these have a lot of mutations that's going on here. Not too much, but there is some mutation, and it may not affect the function of these viruses compared to the consensus sequence, but it will. And this sequence is for the E protein that's part of the spike protein. The E protein that allows these viruses to come into your cells. So you don't really see much mutation there, but it is there. And when you break down a lot of the other, sequences or different parts of the sequence, different sites of this gene sequence from number 1 all the way down to like 3,278. It's 3,278 nucleotides. You see that there are differences. There are a lot of blue, a lot of black, a lot of spaces and gaps. So this is a lot of changes that have occurred. Primarily, I would say in this section right here of this spike protein. Then you see the yellow or gold, I'm not sure, I guess gold color, and this green color here. Now you start to break down, because all I did was take this section here and this section here and blow it up. That's all I did. Okay. And so you see the blue mutation. These are mutations that are occurring. And where you see the red nicks is where you have a lot of mutation that's occurring or that's expected to occur. Where you have these black, sometimes what they'll do is the black will represent a point mutation. The blue will represent like an insert or inversion type of mutation where something's inverted. But here I don't see an inversion. I just see that there is a mutation here with the blue and with the black that's here. And I do see some mutations compared to the consensus sequence. This protein that we have, we're looking at protein now. Sorry about that earlier, but we're looking at protein. We have Wuhan, we have 2019 novel coronavirus USA AZ1. Maybe this is Arizona, I don't know. But it says 2019 novel coronavirus USA. Bat coronavirus ZC45. Bat coronavirus ZXC21. SARS 60, excuse me, 6. Is it GZ02? And then we have SARS from 2003, and we have consensus. So these two are probably from 2019. Wuhan, human, maybe a human patient. And this 2019 USA, probably from another human patient. So they've already sequenced this for people to see this. And this is publicly available. This gives a step-by-step way that they are suggesting they could have developed this particular virus and how it could have been manipulated, and it doesn't really take that much time to do this stuff, by the way. Really doesn't. Like, I would say maybe 3 months or so. It doesn't take much time. COVID 007. Yeah. They never. Shop Talk said, sounds like Al Pacino and Devil's Advocate. They never see you coming. Yeah. I think that's how it works. But that's not a natural way that things should go. But it does. It is happening that way with COVID. Q497. I just looked at a video by BGS called COVID-19 long haulers. Is that what? I don't know. I have to check that out. Q497. I'm not sure. But yeah, we go on. This is step 1, step 2, step 3, and step 4. And of course, this is very small, so you guys may not be able to see it. So I'm blowing up each step to make it bigger. So this is the genome of the template virus. They may have used, ZC45 or a relative virus to that. You can see the open reading frame 1A. You have RDRP, which is required sequence that's needed for this, dealing with the RNA binding motifs. And in another section that I'm not sure about, but all of this purple section is the open reading frame 1B. You have the spike protein S. You have 2 components of that, S1 and S2. So you can take this part right here, excise it out of the virus, and do RT-PCR or DNA synthesis followed by PCR. You run out PCR gel. Simple. You can this right here, this little part right here can take 1 day or less. I could do that and then go to lunch and then come back and run the gel. That's it. Okay. Then the next day, come in, restriction enzyme digest. So this can take probably a day or two, where you have the virus, you First of all, you run the gel, you do the PCR to verify that your virus is what you're thinking it is. And you cut out pieces of the gel, parts of the gel that have the bands, you cut those bands out, and you purify that piece of DNA and put that into a vector, another virus, or you try to grow it up in bacteria, whatever you're going to do, transform it in bacteria, whatever you're going to do. Any way you look at it, you're going to have to do restriction enzyme to cut out the piece that you want and then glue it back together. So that's all this is saying. They introduced EcoR1, which is a restriction enzyme, introduced BSD1, or excuse me, BSD2, BSTE2. So these two are restriction enzymes specific for this virus, ZC45. EcoR1 cuts here at this part of the sequence, which they already know, they just didn't show you here. BSD2 cuts here, this part of the sequence, and when they double digest them, you have a cut, you have an open here. So you can't have a vector that's open. It has to be closed. So what are you going to close it with? Well, you can design an RNA binding motif to close up the hole. And you can ligate them together. You use ligase enzyme to glue them back together. Because you can't do nothing with this. This is going to go in a cell, and the cell's going to chew it up, spit it out. It's done. So they designed through a structure-guided design system. You can see it online for free. They designed an RNA binding motif, back translated that into DNA synthesis, and they used the EcoR1 BSD2 to cut that up, digest that. They put that little tiny piece into this vector. So you have a new sequence you developed from this. So all they did was take out this orange part, which is right here, this orange part of the spike protein that would code for the spike protein. And they put this yellow piece in. This is what they want to go in there. Now you have a new vector. You have a new virus. That's it. Very simple, very, very simple. You know, thank you, Go Professor. He says, is it possible for COVID-19 to be transferred to a child and the child become a carrier, something like sickle cell? It's not transferred like that. Sickle cell is a haplotype deficiency that occurs. It is inherited. COVID-19 is not, well, I should say, we don't think COVID-19 is something that you can have from that you can inherit. It's communicable. So, for instance, there are studies that show that COVID-19 is identified in semen. So a man, if it is in his semen, he would not be, so-called passing it down to his child in their genome necessarily, but it would be, the child would be infected, would have that infection. So basically the child would just have pneumonia if the embryo develops, the child would just have pneumonia in utero, and that would be an obvious issue for the mother and the father and the child. To have pneumonia in utero, that is not good, obviously. But something like sickle cell, no, I don't think it is. So I'm gonna go to this next slide, which shows us, I just said, let's back it up. Let's explain, hold up, wait a minute. What did Nicole just say? Nicole just said, yes, you can produce this virus, a new virus from an old virus. It's cut and paste. It's all kinds of stuff you can use to do it. It's very cheap. It's easy to do. The process I just described here can take like maybe 4 days. And that's it. Depending on how many types of RBMs you want or whatever you want to put in there. I mean, maybe a week or two. That's it, maximum. So what's the difference between a motif and a domain? This goes back to what Name was asked earlier as well. Motifs are similar 3D structure conserved amongst most different proteins that serve as a similar function. Domains are the region of a protein that has specific function and can usually function independently of the rest of the protein. And this is just giving it differences, a motif versus a domain. So I need to get my charger real quick, but hold on 1 minute. So there is a difference between a domain, a domain and a motif. So a motif is a chain-like biological structure made up of connectivity between secondary structural elements. So for instance, that would be like the alpha helices, beta sheets that you look up, and you can just Google the word alpha helices or alpha helix. Helices is the plural form of that word, and beta sheets. And the other side of that is you have disulfide bridges, ionic bonds, hydrogen bonds, covalent bonds, things like that. So that's the domain. So if you want to do drug development, I think that Dr. Dunbar mentioned this on one of his videos. If you want to develop drugs or you want to understand how these drugs are working, you're looking at domains. I'm a motif type of person, so I'm just going to tell you right now, I care more about this side of the protein discussion, but a lot of the pharmacology, toxicology, and the radiology, a lot of them, they look at this side of it more. And see, my thing is at the bottom is very important to remember. The motifs are not stable independently. You have to add something to make it stable. Whereas the domains are independently stable. That's very important for drug development, drug discovery. To have a stable structure, protein structure. Okay, so that's just breaking that down. And as I said earlier, you all email me if you want a copy of this PowerPoint. Hopefully it is helpful to you. If we move forward, they talk about step 2 and step 3. Step 2 is where you take your new and improved vector, your virus that you developed, and you start to put other things in there that you like. Let's see, Shop Talk, he likes this sequence. Kendra like this sequence, BGS like that sequence. Okay, y'all give me your sequences. I'm gonna put it through the website. We're gonna get something out. We can get it delivered here, and I'm gonna put your favorite sequence into this virus. That's pretty much how we do it. It's pretty much how it happens. So what they did, they decided to put a furin cleavage site of the spike gene. Here it is, furin cleavage site, this little green piece right here. Let's just put that in there. Let's make this virus as hardy and healthy and dangerous to the host as we possibly can. That's what they did. You have a primer pair, 5' to 3' in, and then 5 to 3, 5 to 3, because this is a mirror image of itself. So the sequence of this green part. The sequence of this green part is what they're making the primers for. So they already did that. Quick change PCR is what you just do just to check to make sure the sequence is right. It is what you're putting in, and it is. They do the PCR for it to check again. Once it's in the vector, do PCR again. They know it's there. Now they know they can put a certain concentration of this virus into whatever cell they want to put it in. 1 microgram is standard, but I've gone up to 5 microgram of 5 microgram of viral DNA in the cells, or 1 microgram of viral DNA in the cells. That's the basics of CRISPR, basics. And this is not even using CRISPR technology. This is basic technology from like 1997, 2005, somewhere around there. Okay, the third step is using this type of sequence, this RDRP sequence of the RABT coronavirus 4991. I do PCR to get that fragment. Other fragments of the open reading frame, they do PCR. PCR means you're amplifying this part of the gene. That's all PCR means. PCR is polymerase chain reaction. So you are synthetically copying sequences millions and millions of times. Then they did Gibson assembly, which is a standard way to reverse and assemble these sequences back together so it makes sense. You assembled it back together, and now you got the assembled open reading frame 1B gene. Gibson and Sanger sequencing, these are, historically known for helping you to assemble your gene sequences. I mean, you can do this on a computer now. All you have to do is know the PCR sequences, put those little sequences into the software. Some of the software they have, they'll assemble it back to you and tell you what kind of gene it is, or give you a guess. So those are step 2 and step 3. This is step 4, where you have your spike protein. You want to use it and everything. Your full-length viral cDNA genome. So you went from RNA to cDNA, and you did that because you need DNA to infect cells. RNA for this artificial group, thing they're doing. RNA is as we know, this virus is RNA based. But in this laboratory, they wanted to use cDNA, and that's what they're using, a full-length viral cDNA genome. The way you do that is you take a single-strand RNA, you add reverse transcriptase enzyme to the RNA, it copies itself, and now you have double-stranded DNA. It's a complementary DNA strand. So it is seen in a cell as cDNA, as real DNA, even though it's not. Okay. So these 4 stars here indicate very important steps that they did, and all it does is summarize the pictures that I was talking about earlier. The most important thing to remember is that the RNA-dependent RNA polymerase enzyme is extremely important. And if you didn't have that enzyme, none of this would happen. None of the steps would happen. Or it'd be very, very difficult to get it to stay in a stable state. So I want to say that I'm done with my presentation. Oh, you're like, yes, I'm done, y'all. I'm done. I thank you for watching this presentation I've given. I appreciate everybody in the chat. Thank you so much for those who gave in the super chat as well as the Cash App. As always, I'm humbled and I appreciate your support, advice, any constructive criticism and guidance that you can give me. I have my social media shown here. If you want to contribute, you can do that. Every little dollar counts. It's cool. And I have my contact information here if you want this PowerPoint once again. And this is an example of why you don't let 6-year-olds suggest your in-screen, your in-screen. But when you let a 6-year-old tell you, well, I like this for the in-screen, this is what you're going to get. And that's fine. It's cool. But yeah, when you have little kids, little babies telling you, that looks like a good picture, this is what you get. So I appreciate y'all. We'll look at the Shop Talk Live, the test. When is the test? The test is in 2 hours, honey. So you better go study. You could be in there sweating. Oh, it's no problem, India. Oh, thank you, Nameless. Why you say no? Oh, he's saying no, he don't want it to be over. Yeah, this is what they yeah, that's what you get when you have a little 6-year-old. You ask them, well, what you think about it? What kind of suggestion you what should I give? They're like, oh, I like this picture. This little bear is so cute. I just want to hug him. I'm like, oh Lord. Okay, put it on there. So yeah. You said multiple choice? Nah, Sachi, it's gonna be all essays, man. All essays. I'm just playing. Thank you, Go Professor. Once again to you for the super chat. Everybody that's giving in the Cash App, thank you. I'm trying to go back up and catch the super chats at least. I know I wasn't able to put any of the comments in there. But let's see. I was trying my best to just like show the super chats at least, and if anybody wants to come up, that's cool. I don't want y'all to feel like you have to be, you have to, have certain kind of knowledge of what I'm talking about. Like, we all still learning. I'm still learning. I'm just hoping that, any information I convey is helpful. That's all. So don't feel any kind of way about like, oh man, I don't know what Nikole talking about, man. No, half the time I'm still figuring this stuff out. And I have read so much, and the things that I show are based on, they're things that I have read, and I'm looking at their data. I can see the limitations of some of their experiments. And I'm like, I ain't showing that because y'all didn't know what y'all was doing, and I'm gonna keep it moving. But there are a lot of studies out there, and Dr. Dunbar knows this, and many other people that read these types of studies know that they have collaborated. It's not like it was 3 or 4 people doing the work. It's up to 10, 20 people doing the work. Okay, no problem, Nameless. He said he's on his way home now. Okay. Yeah, I just feel like it's best to give you all what they don't give elsewhere, so that you can think on not only my content, but everything else that you're getting, like compare and contrast and see what makes sense. Maybe something I said doesn't make sense. Okay, we can talk about that and figure out, help figure out a way to help you get more understanding. Yes, Dr. Dunbar, absolutely. He said often we have to learn and relearn. Oh my God, if I'm not doing that right now, that is a big one. We are learning, relearning. Like, I was relearning earlier today during our meeting, because I was giving my cells a certain concentration, and somebody said, well, it was like 10 micromolar. And they were saying, well, I read in a study using some cells that they actually give 3 millimolar. I was like, what? Now, mind you, 10 micromolar is, that's 10 micromolar. 3 millimolar is 3,000 micromolar. I'm looking like, huh? Like, I'm not about to do that. She was like, well, that's what they suggested. I said, okay, they suggested that in what way? It all starts with questions. Learning and relearning, you have to ask questions. So I asked her, I said, okay, what type of cells were they? And I'm gonna get you in a minute, Go Professor and Chaos. And I asked her what kind of cells they were, and I'm using prostate cancer, of course. And she said, oh, this was melanoma. I said, okay, you do know melanoma is more aggressive. It's more resistant to some of these drugs that we're using. So she's like, oh yeah, that's true. I said, okay, so that's why they use 3,000 micromolar versus 10 micromolar. If I add 3,000 micromolar, it might kill my cells. So, and then she was like, well, you could just try it. No, you could just try it. I'm not trying to kill my cells. But I will look through that paper and see if it's necessary for me to try that, because that's not a trial that I want to do with cells and then I lose them. I don't know what the heck happened. Now, if they just flat out die, that's different than if they, if the numbers just go down. But if you have a whole plate of cells and they float and they all dead. So yeah, just communicating with each other, learning from each other, and seeing, is that a good idea or not? Very, very important to do. How y'all doing? What's going on? I can't hear you very well.
Come on, I'm on Bluetooth. I don't know if it's clear enough.
You might have to turn that off.
Shit. Hold on a second.
Can you hear me, Nikole?
Yes, I can hear you.
Oh, okay. I wanted to go back to the question I had posed earlier. I use sickle cell as kind of like a just an example, because I couldn't really think of nothing. But if the child has somewhat, like you said, pneumonia during utero, I think that's what you said.
Yeah, in utero.
Yeah. So after the child is born, wouldn't it still be in the child's system though? Does it?
Well, that's if the child is allowed to continue to term, because that is not hereditary. That is not a hereditable disease. Pneumonia, you don't inherit that. you inherit sickle cell. You don't inherit COVID-19. It's just like if the father had, for instance, I'm not sure how true this is. I'm just, going out the fly. If the father has gonorrhea, and if gonorrhea somehow is in the, or what is the other one called? There's another STD out there, but if they had syphilis, that's what, not gonorrhea, I'm sorry. If they had syphilis and then the father, without knowing he has syphilis or some kind of way he doesn't know, but it's in his semen and he passes that semen into the birth canal of the woman he's with, and she gets pregnant and the embryo survives, for a while. Okay, now the child's infected, not carrying that trait. You don't carry the trait of a syphilis or gonorrhea. You can carry the trait of asthma. You can carry the trait of sickle cell. You can carry the trait of HIV. HIV, there's children that are born with the, not the trait, you can be infected with HIV, but you're not going to carry the trait of HIV. You understand what I'm saying? You're not going to carry the trait of some of these. These are communicable diseases versus a syndrome or an illness that is hereditary. So those are two different things.
Yeah, okay, I get that, but couldn't it be viewed as a trait?
No.
I mean,
no.
I'm just trying to, because it seems to me like, things over time turn into a trait. I mean, help me. I'm just asking. I don't, I'm just looking for understanding it is,
No. Not to my knowledge. That I'm just putting something in there. Thank you, American Negro. He said 3rd degree. Okay, 3rd degree burns. Okay, what's going on? I see you here. Thank you for that super chat. Not to my knowledge, you can't have. There are two different things that we're looking at. So we're looking at something that's hereditary, that you get from your family, you get from your parents, and a combination of genes you get from mother and father, versus something that's communicable. SARS is communicable. If SARS was hereditary, why would we wear masks? You don't wear masks for something that's just hereditary. It's just, we wouldn't be having, there's no pandemic if there's something that's hereditary. It's there. It's always there. It's always going to be there. This is not hereditary. You can't hypothetically produce something in a lab that is hereditary. It's produced in a lab. That's a naturally occurring thing.
Okay. I mean, I'm asking. So I understand the trait, thing. I'm just curious, how it would work, because as I said, it seems to me like over time, if something is continuous, it becomes a trait.
No.
I mean, okay.
How do you, so let's walk you through that logic, and how do you think that's the case? Why do you think that?
Okay, so it's just like we go back to syphilis, right? And we remember that there was a lot of testing with syphilis, right? Mm-hmm. So they let people go with syphilis, and certain things happen, but they couldn't necessarily fix or help all the ones that had syphilis, but those ones who had syphilis still produced children and things like that. They may have the child might have been affected, but I guess my question is, doesn't it over time your body becomes I may seem to I'm kind of struggling with what I want to ask you.
Mm-hmm. Take it down.
But I'm thinking, like, because something has been in the person's system for so long and it's been passed through, and then that child is, has some line of it.
No.
Or okay.
This is okay. So when I was showing earlier in the PowerPoint, like the first 3 or 4 slides, I was talking about the timeline and where they began to recognize the coronavirus from the bats, from animals, how there was not much of a sequence similarity between humans and bats for that SARS-1. Not this one right now, but the first SARS. So exactly, Sachi was saying it, and I think Nameless said it earlier. Like you can't. Even with the bats, there's a vector for disease. The vector for the disease is that virus, SARS. For the disease of COVID-19. COVID-19 is coronavirus infectious disease 19. Infectious disease is not inherited disease. SARS is the virus, SARS-2 / COVID-19 disease. They put the virus and the disease in the same name, except on TV they keep showing COVID-19. They're talking about disease. I'm talking about SARS. That's why every time I have it on my slides, it says SARS-2 / COVID-19, because I'm talking about the virus primarily. Viruses, you're not. It's a virus. You're not going to inherit a virus. Now you will have symptoms, syndromes, diseases, illnesses, whatever else that you have that goes away from the norm. A normal, healthy person, that is still not inherited. You can inherit certain bone issues from your parents. You can inherit, like you said, sickle cell. That's a perfect example of something that's inheritable. You can see that in the genetics. You can inherit certain traits. For instance, the Chinese, they did a study on the Chinese and they wanted to know if they are a separate, unique group of people on the planet, and they compared their genomes to the genomes of people from South Africa, and they were showing that many ethnic groups in China derive directly from Africa. That's genetics. So they probably inherit diseases, certain not diseases, but disorders and stuff. I mean, sickle cell it says sickle cell disease, but it's really like a syndrome type thing. But they inherit a lot of those issues from their genetic problems from the people in Africa. Like you see that they have very similar, well, if you look at the inherited things they have, they're similar. But that's not the same as an infectious disease. If I have a cold, if I had the flu and I sneeze and cough on you, like you get it because I gave it to you. I didn't birth you and you had it. Your mother didn't have you and you were born with the flu. No.
Yeah, yes. I get
that. Yes, AIDS is a syndrome. Liang Bentley. So you have HIV virus and you have AIDS, which is a syndrome.
Okay. Yeah, I was just curious, how, it worked. I mean, as I said, I'm asthmatic, so I inherited that because my family, it's in my family like that.
Yes.
So I get it. Some people I know have sickle cell. Yeah, I get it. So with this COVID, I think we're still learning a lot about it, and I that's why I ask. Like, okay, let me ask this question. I'm not a scientist. You know what I mean? I want to know.
Yeah, no, it's okay. It's fine. It's just people will use that in defense of other behaviors that they choose to, take part in. Like, well, it's inherited. Like, I mean, people, no. It's not. There's no way, because like I said earlier, if it's something that's inherited, then everybody would not be wearing masks like this. Like we're walking around wearing masks. This is a pandemic. No such thing that's inherited needs a pandemic, needs to shut down everything like this. People have sickle cell born every day. Children, babies with sickle cell born every day. They don't shut stuff down because the kids with sickle cell. They don't care. They keep everything moving. Gas prices and all kinds of stuff, food going up, toilet paper, people throwing toilet paper, hitting each other over the head, people about to run people over. Like me, I'm trying to cross the street, somebody trying to hit me with their dang car because they in such a rush to go nowhere and rush to go back home. And you wouldn't be doing that for sickle cell. Nobody care about Black folks like that.
Yeah.
They like Black folks got sickle cell and things will continue to go on just like they've been going. No, it is not the same. Sickle cell has all types of foundations, and I mean, just like with Dr. Dunbar talking about the G6PD foundation that they have with this deficiency. That's a deficiency that is inherited. That is not communicable. You cannot cough on somebody and give them G6PD mutation. If that was the case, you can go ahead and finish. If that was the case, they already have done that by now.
Yeah, well, as I said, I asked because you remember when, AIDS came out. Well, nobody wanted to touch the glass you just drunk out of right. Because they thought they didn't want to shake your hand, So I asked these questions because, this is the time now.
Yeah, it's a good question because like Third Degree was saying, it's, great questions. I agree. They are good questions. It is a new virus. It is something that there's still so many things that need to be determined from it and learned from it. But just in general, a virus like this is not something that you would naturally be born with, or, you would inherit from your parents, because you don't inherit viruses, you inherit genes. Viruses are entities, as I said, one of my streams, kind of in my mind, similar to like a parasite, where you have, you're killing the host, but not completely killing them. You're killing them, but not where it's just flat out you're dead in 2 days. Unless that's something that has been engineered. But usually viruses, it takes time. It takes a long time, and it's not. You can't. Looking at the genes that I was showing earlier, looking at the protein sequences, none of that is like none of that would translate into you're inheriting a virus from your mom. No, that doesn't happen. Like if the mother has HIV, they give her drugs to block the immune system of the baby, of the fetus, so that they can coexist together, so that the mother's HIV is not going to kill the child, and so that the mother's immune system won't kill the child, and vice versa. So they give them medications to slow that down and keep each of those entities, the mother and the child, from killing each other. If the mother has HIV, that's why children, like the young lady years ago born with HIV, she was born HIV positive. She was infected with HIV. She does not have HIV in her genome. It has not incorporated into her genome. Now, mitochondrial DNA does have components from evolutionarily does have components of viral genome, but that ain't got nothing to do with COVID. That is from thousands, tens of thousands of years, hundreds of thousands of years if you go by certain studies. Because your mitochondrial DNA, you have two types of DNA in every cell. You have the nucleus DNA, and you have the DNA within the nucleus, and you have DNA within the mitochondria. Nuclear DNA is DNA primarily from the father. There is some maternal DNA there, but mitochondrial DNA is exclusively maternal. It's exclusively from your mother. And that's what they're saying, that that is where they see similarities in virus DNA. That doesn't mean you're infected with a virus. That's from over time, things changing and adapting to external stimuli, external environment. But that doesn't mean like your mom gave you a rhinovirus and you got it in your DNA. No. Your mama didn't give you that. You just need to go blow your nose, honey. That's all it is.
Do you believe that the COVID now will be with us for a while, or do you think it will evolve into something. I mean, I see different information about, different strains have evolved. I'm curious, because, we refer back to SARS. When SARS came out, it was like, the big thing that is happening, and it's happening to many people, blah blah blah, right? Then we end up with COVID. Is it the same lines, or do you perceive this as, okay, this is the equivalent to SARS, it'll blow by?
No, I don't think it's going to blow by. I think it's going to be with us for a while, because it is not the same as SARS. That's why I was showing in the slides. The sequence is different. So the sequences has been changed, perhaps hypothetically, I'm going to say allegedly. The sequence has been changed allegedly to make the virus more, make the virus stronger, make the virus last longer, make the virus go into different organs quicker and stay there longer. And it's not like the first SARS. Like I was showing some of the mouse data that they did this year, that the work they published this year, where they were showing all these different organs that have the virus there, and it's lasting a long time. People who are infected with SARS-2, it's not the way they were with the first SARS, because you have young people with no symptoms of the viral infection who are dying of strokes and heart attacks, or have long-term kidney damage and they have to be on dialysis, or they have some type of bone marrow issue because their immune system is exhausted, stressed out. This is a different type of virus. And I was just trying to explain in that study they were doing that this is allegedly how this virus could be different. This is how it could have made it different at the gene level. But I don't think it's going to be like, oh, well, it's Christmas, SARS-2 ain't here no more. I've heard people say, oh, next year in the summer, in the spring and summer, SARS is going to be gone. I said, this is not a freaking airport, or where planes just take off and land every day, like gone here today, gone tomorrow. No, are you stupid? That's how I feel, honestly. I'm not saying it to you. I'm saying in general, people say stuff like that. Well, when it gets hot outside, they said that this summer, did they not? When it gets hot, Memorial Day 2020, SARS is going to be gone. Okay, go ahead and kill yourself.
Do you
it's
think
not going to be gone? This is going to be the norm. Wearing the mask. I mean, washing your hands and doing that should be standard. Okay, if you're 4 or 5 years old, that's standard. But, wearing the mask, having the distance, we already know the virus spreads more than 6 feet. We knew that since April, people just now realizing that in October. But okay, so we know that it spreads more than 6 feet. We know that we need to wear a face covering, the right face covering. That's going to be the norm. Eating at restaurants, going to bars, going to outside events with a lot of people around. That's not going to be, that's at your own risk. You're going to have to do that at your own risk. If you want to go and have fun, and go to the beach and chill and be with your friends, go to the club and buy these girls drinks and all this kind of stuff, be all up in their face, you might not see nothing in 2 days. You might not see nothing in 2 weeks because you're going to be in the ground. That's just the reality. So you better, and like I'm going to say, like my dad said, these dudes out here better enjoy this pussy out here because they might not see nothing forever. I'm serious. He said that. He was telling me, he said these dudes going to clubs, they shooting clubs up, still shooting. I mean, for real shoot, not like that, but really shoot.
Yeah, I get what you're saying. Yeah, I get what you're saying.
Clubs up with guns, and he was like, they out here going to clubs, you out here, drinking, and he said, that better be the best. You know what? That better be the best girl you ever seen in your life, because it might be the last. The last cat you gonna get. That's what my dad said. He was like, you need to be careful. He was like, this is no game. This is no game. He was like, people out here not wearing masks. We have what was his name? Herman Cain. All these people that's just driving. That ain't no joke. That ain't no play. That's for real.
Right.
This is gonna be the norm for a while. And I just hope that, that we hopefully we've realized that and we're taking the steps to be more careful. I'm home today because I had the flu shot last week and I'm like tired and, I don't have flu symptoms per se, but I have like, the malaise and stuff, the fatigue, the soreness, muscle soreness. I don't have like coughing and not too much. Have some congestion, but that's just the flu vaccine. This is basic every year. I have to stay in as much as I can because I don't want to be vaccinated and then turn around and get infected because I made a mistake somewhere, get infected with COVID as well. Just being careful.
Do you believe, as a person who works in this field, have you seen more of an increase of a financial attachment to this? Is money garnering these? Because I believe that this is a created disease, infection, whatever it is you want to call it. Have you seen financial endings contributing to this, moving to make money off this and continue to promote, this disease pushed out there to garner more financial worth? Have you seen that? Are you experiencing that?
I'm not understanding the question.
Okay, so COVID came out, right? So things had to move differently, right? And then the government and places started throwing more money out there to like people say, oh yeah, we need money for this and that, like say like Kaiser, right? Kaiser wanted to, say, oh, we got this many people for COVID. We need this much money. We need this much money to take care of this. I'm asking you, have you seen more money thrown in the mix to somewhat for people to gain more position as far as monetarily?
Yes, I have. I had an article. I think I might have put it up. I don't remember. But the money that's needed from different countries, putting how much money in the billions and stuff they've put up for COVID research, for COVID studies, for all types of things. I mean, the Department of Health and Human Services has grants and opportunities that are specific for coronavirus. They have a SBA, of course, they have the coronavirus relief options for Small Business Association. But yeah, there's shifts in consumer finance and consumer marketing, how things are marketed. If you notice, if you're watching a lot of the commercials, you see people wearing masks, you see people doing certain things that would be pertaining to COVID during the pandemic, the way people are dressed, like all that kind of stuff, the way people are interacting with each other on some of the commercials. The commercials are there to sell you something, right? So that's all dealing with finance and economy. So they're even whether they're selling you a bag of chips or they're selling you a depression drug, they got people with masks on. They have people who are distanced, and walking and things like that. So yeah, not only that, but in the study of coronavirus, there has been a lot of money poured into it. Of course, like I said, over 3,700 trials. Those trials are not cheap. They go up in the millions for one trial. If you have one trial for 500 people, 500 participants, 250 on each side, 250 for the control, 250 for the treatment in the trial. Plus you might have 200 employees and staff and support staff, maybe 50 consultants. You're looking at $100 million trial.
Yeah.
Easily $100 million. Who's going to pay for it? Not the federal government completely. They might pay a quarter of that, but now you need 75 million where you gonna get that 75 million from? You can go to Pfizer, you can go to Moderna, you can go to, you know, all these other biopharmaceutical companies. You can go to PHRMA, you know, all these different companies where they have the manufacturing of vaccines, where they test it in-house and you study it in your trials. So all of that is money. I have, to answer your question, absolutely yes. What's going on, Tom Malone? I got Nameless here.
Hey, what's happening, Tom Malone? Hey, what's happening? Oofs to Nameless, the protagonist.
Oh, salute, fam. Also, Nicole, thanks for having me up.
No
and I was doing some shopping, running out there and doing my little zombie run real quick. Had to re-up on supplies. You know what I'm saying?
Yes, sir.
You were out there gathering supplies and foraging. That's
right. All my Shinobi no Mono type stuff, man. But,
yes, sir.
But yeah, like this was interesting. It's kind of like I said earlier. It's kind of funny that they're trying to pass this off like some immaculate conception idea, which, and I'll put my tinfoil hat on this. Real talk, from what I see, I think what Nicole is describing as far as the financial incentive is more like a financial reaction. Mm-hmm. That this whole scenario is a grenade that went off way before it was supposed to. It wasn't complete. It wasn't calibrated to where it was supposed to be, but it was further along enough in development to where bad as it could have been, but this was a worst case scenario that occurred.
Yes.
So somebody messed up.
So essentially you're saying they threw money at it, with the hopes that it...
No. Well, let me go back in order sequence.
Yeah.
Let's say you are in the midst of trying to create a economic shift by adjusting the landscape that creates an economy. You want your economy to become the new global economy. So what do? You need obviously destabilization. And there's multiple ways you can make that happen. Now, let's say that you are able to create a invisible disease that is able to target a specific group of people, that follows a specific genome and is almost damn near non-detectable, and can create circumstances that remind you of AIDS where, well, let me use a different example, that compromises your system in a way that creates immediate death due to acute circumstances, and can do so quickly that it's almost like it's a weapon. Mm-hmm. I think that's the intention we're seeing with its design, because when you look at the way gene editing has gone, my red flags came up when I learned about GMOs. But then you look at what went down with the response to the Zika virus, and they began sequencing mosquitoes that had a genetic disposition or a genetic sequence to eliminate the breeding process after a certain number of generations.
Right.
And then introducing that out into the wild with no clue as to how that may mutate. Like when you introduce a sequence that doesn't occur naturally like that, you've got no clue what may respond to that. And mosquitoes reproduce so rapidly, just within a single year. Here you are throwing all of that out there into nature, and who knows what kind of microbes are, responding to that RNA. And just like NikoleAli was talking about, that RNA may not necessarily just be intrinsic to that mosquito. It's taking in blood from other sources. It's passing it on to other creatures, because you got to remember, what do mosquitoes do? They suck blood. You're coming in contact with all kind of different.
They do two things, actually. There's two things. They suck blood, but they also insert anticoagulants into your blood, and they can actually contain the anticoagulant enzyme. Is specific to that mosquito, but also specific to whatever the mosquito is carrying, the vector. It's a vector for, like, if a mosquito is carrying a virus, it's going to be, they have producing that enzyme specific to that virus and that mosquito.
Right, and it's important that it's RNA, and to prove the point you were making earlier, in the cell. There's why they describe that, as being in the mitochondria. that is your powerhouse for the cell.
Yeah.
So there's a little bit of symbology going on there between the masculine and feminine dynamic, but I will leave that to another day.
I know.
You hip, but a lot of people may not be. But you know,
I said it, I was like, damn it, why'd you say that?
But you know, it's profound because it's potentiality to mutate. And just as she mentioned earlier, when you put in that anticoagulant, you know, it's what you use to kind of make sure the blood is nice and digestible. It helps break everything down. You're introducing that into somebody's system, which then, you know, goes on to replicate further. And here you are with this foreign sequence of terminate reproduction after such and such generations. And what do you think may potentially occur? You have no control over that.
You have no control, but you can, you know, a few things are true. You know that you're going to have a, at some point, number one, mixing of this artificially created mosquito with natural mosquitoes. Number two, before that, you're going to have fighting going on between mosquitoes and territorial response. You're going to have them mixing at some point. So you have this chimera strain in the middle. You have one strain that's natural, one strain that's mutated or artificial, and another strain, which is a chimera of the two. And lastly, you have whoever is the best. Natural selection will take its course. More than likely, the natural selection will occur, which is unnatural natural selection in my mind, because more than likely the mutated or artificial genome will take over and the natural mosquitoes will be killed out. Now, the only way to do that is to go through the really reproduce faster. That's the only way they could do it. And that
may make a bad problem worse.
Huh?
Which may make a bad problem worse. They may encourage the thing they were trying to stop.
Exactly. And they'll have this gene in them that is not natural, which could also affect people that are infected by this mosquito when they suck blood. You know, whatever is there, you don't know what, you don't even know if that mutation of the mosquito or that change in the genome, which is a mutation, right? If that change affects the proboscis that the mosquito uses to suck blood, it could actually change the structure of the proboscis to make it longer, to make it, you know, wider, to make, you know, it push more blood out faster, all kinds of things. So, you know, there's all kinds of things that could happen. It could make the mosquitoes fly faster. It's all kinds of things. So you don't know. What the worst thing it could do would be to shorten the gestation of the mosquitoes.
Hey, is it possible? Is it possible for it to come from, like, let's say, horseflies, or, you know, if you're not in a certain region or a place where you're privy, you know, you're subject to mosquitoes, could it be possible that this can come from certain biting flies? Or
okay. Well, I think Namus is going off of a actual experiment that they have done. I talked about it, I think once on my channel, where they were just mutating these mosquitoes and they were going to breed them out genetically. They're going to breed themselves out. And they've done research, and that my friend sent me an article a couple weeks ago about the mosquitoes that are artificial. You know, they're putting out that they're not even real. They're more robotic mosquitoes and everything. But what Nameless is talking about is a specific type. Horseflies are not everywhere. We have mosquitoes, and I mean, you have mosquitoes in the desert. You have mosquitoes everywhere, pretty much. The problem is not the fact you have the mosquitoes. What they want to do is, you know, let them breed out themselves. But it's not only the implications that you're hurting the mosquito population, but whatever would eat mosquitoes are now eating mutated mosquitoes, and whatever, and eventually they'll be gone. And you're left with animals that ate mutated mosquitoes. And if you could do it to mosquitoes, who else could you do it to? Are you just going to go up the chain of animals, of insects? You're going to do it to bees? Are you going to do it, you know, are you going to do it in the opposite direction to produce more honeybees? Perhaps that would be good, to produce more honeybees. You know, how is that working? But I don't think, like, moving it from, you have a point, you know, moving it from mosquitoes to horseflies could, you know, potentially have some some value, but I just don't know how that pertains to this, what he brought up.
So what I'm, just to clarify, Gold Professor, the reason why I brought that up wasn't to really focus on the mosquitoes. It was to focus on what occurs when you take a genetically edited organism and place them into an environment where there's synergetic responses to your environment everywhere. And these interrelated relationships, for a redundant term, don't know how many things were contingent on things being the way they are. In other words, when you make that slight adjustment to something under the presupposition that, okay, this is a problem, which are generally man-made, ironically, you don't know how nature's going to respond. You can't control that. So when you introduce a sequence that normally would not have been possible, such as terminate the generation after such and such generations, that's profound, because you are now putting into play a sequence that nature did not create, not on its own. Because typically speaking, when you see these things form, they have some sort of, they don't form so uniformly and so uniquely packaged to where they can create these nightmare scenarios where if a species goes extinct, it's normally because the environment, one, has changed so drastically that the species can't adapt quick enough, or generally human intervention, which is ironically just another impetus for environmental change. So the fact that they were able to sequence the SARS virus to the point where it can be weaponized, essentially, this is the problem. And just like Nikole was saying earlier, this isn't even loaded yet with other sequences that could be even further tailored to make this worse, which is why I think, again, this escaped from somewhere prematurity. It wasn't complete yet, but it was so far along that they've got the vector set, they've got the way they want it to behave, it's got its stealth mode. It's just causing direct issues and opening the gates for people who already have complications to experience them with, maybe natural progression of this virus producing weaker strains, which I think they're seeing now, but also stronger strains where they're more aggressive breaking down of the lung tissue. And that's why I made that comment in the chat earlier about it looks like, with the lung tissue being jacked up and being loaded with the virus, it's going into the bloodstream. And where's the next place it's going to go to? The heart and the liver and spleen to try and purify it. And what do you do when it's loaded with the virus? It's telling it to, basically self-destruct. It's crazy.
Yeah.
Find that in nature.
Yeah, it goes to what I agree with you. What you said earlier was, I believe it's a created element. It's a created thing, and it's designed to move a certain way. Now, once again, I'm not a scientist or a chemist or anything like that. But what I do know is that this thing has a profound effect on your being, your body, and it's designed, it's like that robot. You've seen Transformers, right? And in Transformers, they have these little robots that land on you, then go into your skin, and next thing you know, it's doing something to you. It's
like
it's making you move a certain
nanomachine.
Yeah. I mean, I just find that it's just like we go to the Matrix. You remember when Neo, I forgot her name, put
the bug that was in his stomach.
It's like that.
It's not a virus, but I understand what you're saying. But
it's not. I'm not saying this. Yeah.
But when we like when we were talking earlier about something that's hereditary versus something that's communicable,
right,
something that infects you, that is not different, because even on the Matrix, when you saw that little, what is, I guess, a robotic type of little machine come out of the stomach, that was they were not born with that. That someone had to put that in you. You're not born with
right.
that.
Right. I
So
agree. And I'm saying
it's harder to treat. That's why we have it's harder to treat, but it's less likely, of course, it's less likely to spread if you're born with it, because humans are not having, as many children as we did back in the day. Even back in the day, if a woman has 15 kids, she can't have them all at the same time.
right.
Well, those hereditary, and if someone has a hereditary disease, they're going to tell you, hereditary syndrome, they're going to tell you that up front, or, that's expected that they tell you that. But someone that has an infectious disease, it is in the word. You're able to infect hundreds or thousands of people before before the end of a week or 2 weeks or something, all the people that you come in contact with. This is why I had an issue with people coming around, excuse me, I'm about to rant, on BGS's channel, on his streams, and they talking crap about, and his name was One Drop from Amit Zia. Yeah, you, honey. You was on there, and you was telling BGS, and you in California, which is really crazy. And BGS is like, why is it in California? Why the cases in California not as high as they are in New York and Louisiana, New Orleans? Because people are closer together. The infection rate is higher. The R0 value is higher. They're walking close to each other. I can't tell you how many times I walk, like face to face, side by side with somebody, even now. It doesn't happen that often. But if I lived in New Orleans, if I was back home where I'm from, yeah, I probably would walk by a lot of people, where I would live. So that right there shows you the infection. Infection is just totally different from heredity. And I put it in the chat. I put a link to something in the chat that explains it a lot better than I can. But yeah, that.
Yeah, I understand what you've made it clear and broad understanding of that. So
Yeah.
I'm clear on that. But what I was speaking to was what I believe that is created, in which that's why I made the analogy to somebody's putting it, created something and put it in, just like the Matrix. They had this thing that went into the stomach, and then later the agents put the thing in Neo's stomach, and then later they took the thing out of the stomach, right? So I'm saying, they created something to put something in you. Now the deal is to find out how to take it out or keep it from getting into you. That's the thing now.
I think it's way more horrifying than what you're talking about. Yeah. I really don't know how to express how much they effed up when this came out. This has been a complete genetic catastrophe because it's airborne. It hides from your immune system and you don't detect it until it's too late. And it operates off the cancer paradigm. Whereas you need to see evidence of its destruction in order to recognize it, or you just roll dice and you happen to just get a serum test that shows, oh, hey, look what we found.
Serum don't work. Yeah, even the serum plasma test for certain things don't work anymore, because if the test is not, that's why Dr. Dunbar was bringing up the BGS, like, the COVID test they were doing, some of the original RNA test, DNA, cDNA, RNA tests they had were not accurate because the threshold was too high for certain concentrations of DNA. And this is not a DNA virus, this is an RNA virus. So that's another step that you have to do to make RNA turn into cDNA, complementary DNA.
Right. So
that's right there. So even if you get cDNA from a low yield of RNA, it's going to be low yield cDNA, right? So when you get that low yield of cDNA, for instance, if I take a cup of water and freeze it, I got a cup of frozen water. If I take a gallon of water and freeze it, I got a gallon of ice, right? Frozen water. So they are not equal to each other. So if I start off with a small amount of RNA, I'm going to get a small amount of cDNA. If I start out with a huge amount of RNA, it doesn't take much to make a little bit of cDNA, because the concentration is so high. However, with this particular thing, they have really messed up. You're right. They've messed up because at one point they couldn't even make primers against the RNA sequences because they realized it was mutated from one patient to another. That's when you start to recognize, okay, there's a mutation, because I bring patient 1 in and I have the sequence that matches this person has COVID. I bring patient 2 in, it doesn't match at all. They don't have the same strand.
That means,
or it has 25% identity, which it means it matches 25% of the time. And that's not enough. That means it's a totally different thing you're looking at.
that means you can have so many different forms you've got to calibrate for just to even be sure you're dealing with it with something so time sensitive and how long it takes to verify and confirm. This is where you're getting a lot of the false positives and false negatives. It's this is you can't treat this like the flu because it behaves in a way that really it's abnormal for virus behavior that occurs naturally, which again hints back to what NikoleAli's highlighting here. The behavior is so anomalous that you can't ignore this. Nothing in nature acts like this so conveniently.
So let me ask you guys then. You know, I'm curious. So how are they testing for something that's so evasive?
You remember that they're doing it in a very painful way because the original tests they were doing were based on the original SARS. So they were doing the tests like I was just saying, the RNA type of test, but the nasopharyngeal swabs that Superior's people, nurses like him, doctors that would do that. He said on Saturday, Dr. Dunbar stream, he did nasopharyngeal swabs. The NP swabs came out negative. They did the deep bronchial swabs where they go deep down in your lungs while you are awake. More than likely, you're awake. And that is extremely painful. And they go down in your lungs and take samples like that. And then you're positive. They noticed that's positive. He couldn't tell us exactly what the concentration of the titer, how many viral particles were there, but he just said he gets the test back. The results will come back to him. It says positive or negative. It says COVID-19 detected or COVID-19 not detected. So that's one thing you can do. One thing they have is the deep bronchial test. They have the nasopharyngeal test. They have the blood test, which is going to tell you your timeline, basically, of your infection, how long you've had it. They can give you an idea how long you had the infection. And then they also have, one thing they were saying they were doing was the cheek swabs. Because if you take cheek swabs, you get DNA from that directly, right? Even though we know it's an RNA virus, you get DNA from the cheek swab cells, those epithelial cells inside your cheek. But if you sequence for certain genes that will be high during a viral infection, like SARS, then they know you probably do have SARS, at least a SARS, a type of coronavirus. They won't know specifically if it's SARS-2, but they'll know that you have these genes are up, you probably have a coronavirus, So they have different ways of looking at it, but even then, it can change within a patient.
Which is really interesting and kind of telling, and again, supports what you were talking about earlier, about how, even for checking for one, there's so much damn overlap and plagiarism going on with COVID that it kind of implies the other through association. It's really hilarious.
Mm-hmm.
It's hard to defend this immaculate conception with COVID. That's what I'm gonna call it now. This is the immaculate conception theory.
Who is
COVID-19? I mean, doesn't it encompass what they're trying to pass?
I know what it is. They just wanted to be Christmas all of a sudden. They want Christmas to come right now. I want Christmas to come like last month. I just want to hurry up. Well, you can't. So
right.
But let me talk away real quick. Shout out to Dr. Dunbar for the Cashapp MLR, Ron Reed. Let's see, MLR again. Okay, Ron Reed. Shango. Thank you, Shango, for the Cashapp, and Vernon King. Thank you so much for the Cashapp, everybody. And hopefully, in the future, I'll be doing, in the near future, I'll be doing something else with Dr. Dunbar. So hopefully, we can get that together. It's going to be possibly on the G6PD deficiency, because I think that's very interesting to look at. It could actually fold into something that's that's pertaining to like legislation for people who have that deficiency, similar to what you see with sickle cell and cystic fibrosis and other issues that people deal with, asthma. I think that G6PD needs to be more on the national stage because it affects so many African Americans and puts you at a disadvantage for a viral infection such as COVID-19. So it is not listed as a potential risk factor for COVID-19. So I think that's one of the first stages, is to get it established as a risk factor. A clear risk factor for COVID-19 would be, in future infections and mutations of COVID-19 would be the G6PD deficiency. Meaning, if you have this mutation and there are over 200 mutations of this gene out there, if you have any mutation of this gene, you should be treated with specific care.
Wow.
Because it affects your blood cells. It induces anemia, hemolytic anemia type of symptoms, and so that's the next project, hopefully, that Dr. Dunbar and myself can work on. And I'm just so happy that I can come along for the ride. So we'll see what happens with that.
That sounds cool, because then you can get into a preventative culture with some of this, at least, because again, I think the best thing everybody should walk away here with is, one, you can develop antibodies to this, which, that I hadn't even heard that. That's huge. And if the antibody treatment is actually helping to address that, that may be a better way of dealing with it, to at least reduce the probability of you being affected by it that way, especially since we know that the pre-existing conditions of the body seem to be the common denominator with, the mortality rates with a lot of this stuff. So, this may be another good opportunity to really push for generalized health with regards to this.
Thank you, Top Talk, for the super chat. He said, "Thank you for the instruction." Oh, it's no problem. I'm humbled that y'all come and listen to little things I have to say here and there, and I do what I can. But it's not really going to be, in my opinion, it shouldn't be for a generalized healthcare or health upon this, within the health continuum. It should be more towards precision medicine, specialized precision medicine, because everybody else that has issues, certain communities, certain ethnic groups that have these different types of mutations, they focus on personalized and precision medicine. I think the same thing should be true for the G6PD deficiency and the different mutations that occur, because most of these are point mutations, meaning that you have exchanged one amino acid for another, for instance, or you flip base pairs around, instead of GATC, it'll say GACT, for instance, so that's why I'm thinking that would be the best option, because everybody else is moving towards, like even in my laboratory, we're moving towards precision medicine, personalized for each patient, because that generalized medicine is what we've been doing for a long time, and it has not worked since the beginning of this country. Since before the beginning of this country, generalized medicine was what we did. Since the beginning of the Spanish flu, since the beginning of the bubonic plague, they had generalized medicine. Everybody had the same thing, and it worked, ? For most people. But we also didn't have the diseases brought on by the environment back then as we do now. Not as many, as far as heart disease, hypertension, diabetes, and so forth. So those are risk factors not even risk factors. Those are critical care notes to keep in mind when you're dealing with the G6PD deficiency. Why? Because those are the same types of things they use as a critical care note when they look at other people, such as cystic fibrosis. They look at PKU syndrome. They look at, some other things that would affect, different ethnic groups. That's how they do that. These are critical care notes that they make sure that they understand, AKA comorbidities.
Yes, and I just want to make it clear that when I was making mention of that, I was talking about, how they could take the plasma samples, extract the antibodies, and start really focusing that as a general treatment to use in conjunction with recognizing groups of people who may have those genetic mutations that leave them more susceptible to this, like you're talking about, so that we can focus on those targeted treatments. And that's a really big dynamic that's been expressed with a lot of the homeopathic or the natural and the herbalists, that circle, having that specialized treatment based on known issues dealing with your demographic, and then furthermore dealing with you specifically. I know Kaiser has been trying to really push that sort of dynamic with a lot of their palm colored patients, because I see them have conventions on this quite a bit. My mom is a psych nurse, and even in that field, they're getting more so into the specialized psychological dynamics with regards to, inherited mental issues that go on that way. And it seems to be the direction that medicine is going. But again, we go back to that same common denominator problem of where's the funding? So it is a blessing you guys are having these conversations to, one, spread awareness of it, two, demonstrate that you do matter on an individual level, and you do belong to a demographic, and no, your issues don't need to have some sort of trickle down blanket statement adjustment, because our issues aren't the same.
Absolutely. That is what you just said. The last thing you just said is exactly why the precision medicine field is exploding right now, with everything developing with tech, things being incorporated from other disciplines, such as like what you mentioned with psychotropic and psychological genetics and everything, looking at things from a pharmacokinetics standpoint, which is what Dr. Dunbar focuses on, is how these drugs are affecting your body. But the last thing you said, like, this is what it is. Like, we're going forward. You have to focus on people. You are important. We have to know what people need, what you're coming to the table with, what you need. We can't treat everybody with a blanket. It doesn't work. Everyone with the same blanket. No. And then everyone will, have issues. Everyone's not going to have the same response. Everybody's going to have issues because everybody doesn't fit that one blanket, that one package. And this all goes into some of the things that the Black Brain Trust talks about as well, because, for instance, with the universal basic income, you can help alleviate some of these problems, some of these risk factors. If you had universal basic income, and that's not just giving people money just to give it to them, because I've heard people say that, and I'm like, okay, you didn't read this, this concept that is being used in different places as, experimentally, but it's as a preventive measure. We can prevent you from going through this psychological, downturn due to financial troubles and woes. We could prevent a lot of these issues. Of course, we know that in a lot of cases, your ability to eat healthy is due to socioeconomic factors. We know that. A lot of places where people don't make as much money, these are food deserts. If they made more money, would they buy food? Yes. Mike B talked about that. I think it was over 40% people that were given UBI in the test environments, they spent it on food.
Yes.
People know their position. They know a lot of times they know their health issues. They want to do better. They do have a utility for the money that they're given. It's not just giving money out to hood rats and broke dudes on the street. No. That's not the focus. That should not be the focus. But I'll go ahead. Go ahead.
Yesterday, my cousin, I found out, died of a stroke, and he'd been dealing with a lot of stress from his family. His baby mother essentially was running gynocracy.exe. She was a whole drag, damn near kidnapped his kids and ran off without saying nothing, demanded child support. All of this stuff was going on within the last year. And so, he ended up he had a few, and I think it was probably connected to COVID. But I remember in the time that I spoke with him, before I moved out to Texas and all of that a while ago, he did have issues with diabetes, and he just wouldn't change his food habits. And he looked at how I was eating and was just like, I don't know how you do it. And, I broke everything down to him on why he needed to address his food habits. And my whole family's like that. And it was only the people in my family that started taking their diet very seriously and started going out of their way to try to control that even one, don't have diabetes, don't have high blood pressure, and aren't on medications to try to address these things. So it's very real. The stress does play a role. And at the time, his ex-wife showed up at the hospital, and within a few hours after she left, he had a stroke and died.
Oh, wow.
So, their conversation was about, the rent that was supposed to be due. And, I I found out a lot about this stuff. Like, my mom called me again, and in the same conversation, let me know my grandfather actually died. I didn't know that. Same deal with him. He had gotten COVID, got over it. And then, he was trying to get back into dealing with the whole fitness thing. And, he ended up having a stroke too, 2 weeks ago. So
Yeah.
I'm just finding out all of this last night and how it's connected to a lot of these things, which is why I was trying to say, this is hitting home for me right now, because it's affected me and people I know directly. And there are components that are connected to lifestyle. And there are things that are very common denominator that affect Black folks disproportionately, like stress and things directly connected to our food habits, which again imply socioeconomic status and the culture that gives you your blueprint on how you put food together. But I didn't want to go too much on a digression on that, because that may warrant.
No, that was absolutely necessary. That was necessary because a lot of people don't understand. They don't understand, like, this. Just it's just so it seems like it would be so simple, right? But, and my condolences to you, absolutely, because that's a hard loss right there. But people just don't understand, like, just if they just look, first of all, if they just looked at your IG, I mean, just like create like a little cookbook. Well, it's gonna be a binder of notebook paper of my notes of your recipes. Like, if they just that, if you just like made some videos or little clips or something and show your family members, that would be great. I mean, and the fact that you're talking about it, you don't know the words you're saying could be a warning to somebody, a revelation to somebody out there who may be like, you know what, he's right. I need to make a change. And that stress piece is very important.
Mm-hmm.
Like, you can literally feel like your blood pressure. People are like, I feel my blood pressure going up. That is not good. If you could feel it going up, that's not a good thing. You got to work on that stress management part of it. That's very, very important. But I'll let you continue if you had anything else you wanted to say to that.
Oh no, the doctor came up, so salute to Mr. Dunbar.
We gotta be quiet.
What's up, y'all?
Salute.
Lioness, nameless.
How are you doing?
I'm doing all right. Speaking of stress, I'm trying to refinance my mortgage and stress. But just piggybacking off what you guys were just saying, I was thinking one of my buddies who's not a firm believer in COVID-19, I just realized he's letting himself slip, and he's gained a tremendous amount of weight over the last couple of years. So I just thought about that. But BGS actually tagged me in an article about the convalescent plasma. So I recorded a video on it, but I have to finish it, and I'm probably going to upload it tomorrow. And as you guys were talking, I was thinking that in that article, they talk about people who have created a robust amount of antibodies. So those people might hold a secret to beating this thing, because I'm sure they're researching those people to see, what's unique about these people that have allowed them, excuse me, that has allowed their immune systems to rebound in such a strong way. So I think that's going to be another area of interest.
Yeah, I think the neutralizing antibodies, the IBO7, and it's just a class of neutralizing antibodies that they're using. I think the sequences of those antibodies were developed from the convalescent plasma antibodies of the patients. So that's another reason why the study was so good. I mean, there were things in the beginning of the study that were very depressing to me personally. And then as it went on, I just kept going. I was like, where's the good news? That's why I kept saying, don't bring me no bad news, because while I was reading that study, I kept getting emails from people like every other hour. It seemed like I was like, stop bringing me bad news. I felt like Eveline or something. But when I looked at the end, I said, oh, wow, this is great. This is awesome. They have a good plan. And they developed this from, the synthetically developed neutralizing antibodies. What they do is take the sequence of antibodies produced by patients with the convalescent plasma. They find out what those antibody sequences are, the protein sequences, and they can synthesize them, give it intranasally to the mice and, in an aerosolized little package. And then you have this reduction in viral load over time. So that was good that they can use that. So that's just an example of, a sterile version of the antibodies being used. They're getting the sequence of the antibodies and the proteins from the convalescent plasma. They're not using the plasma directly because, of course, human convalescent plasma has no effect on mice, right? Well, it shouldn't have an effect on mice, and especially mice that are already COVID-2, SARS positive. They're not going to have any effect with this human plasma. So they had to develop it synthetically. So even with the synthetic sequence of this antibody, they see a drop because they got synthetic virus in their body, but there's a clear infection rate that's happening. So the mouse immune system thinks that there's a virus there, and it's just acting on it and trying to, not much because it's, compromised, but it's doing what it can to try to help these mice survive, and they're eventually dying. But before they die, they give them this antibody and they get better. So yeah, it is a good model. They said that many times in the paper. It was a good model for using convalescent plasma in humans and a model for reducing viral load that is extremely high. Because in the mice, that was very high. So yeah, I mean, the first time I went through it, it was, I didn't get a chance to go all the way through that study, but I was like, let me try to, reintroduce this because there is a good end to the story. So.
Yeah.
Well,
Go ahead, Fab.
No, I was just thinking, as you were saying that, Nicole, Nameless, I heard you earlier on in the stream, you were talking a little bit about evolution and things getting killed off. This thing might be, I hate to sound morbid, but this thing might be, I don't know, weeding out certain populations of the population. Yeah.
Yeah, I mean, look at how it's tailoring things, right? Like, let's say, and that was kind of that's the energy I sense from the direction this is going, where if you were about to take over the world on some big coup, right? Let me put my tinfoil hat on. You get rid of individuals past a certain age. You get individuals out of here that are already compromised that could bog down the healthcare system. And you leave individuals that are malleable age-wise, but not too old. You can sweep in and easily take over.
Yes.
So, and that's the end of my tinfoil rant. But when I look at this at the end of the day, it's why I say, okay, if you understand that people who have pre-existing conditions are going to be compromised, you absolutely have to take your own personal health much more serious. There is no hiding from this. This is not the flu. This isn't going anywhere. This was not immaculately conceived by nature. Somehow this behaves entirely too much like something that's been given directions and interestingly tailored to the human body. You need to take this serious and take advantage of all of this available information of the vectors that it travels through, the way it works, and why it works. Do not make it easier for this thing to continue to do what it's doing, because so many of these deaths end up being self-inflicted before we even run into the real problems, which is the healthcare workers and the healthcare providers not being able to provide help for those who need it the most. That's where things are really going to get bad if they do. And the supply lane people and the healthcare workers, those are our two bottlenecks. If that goes down, your lifestyle is going to be a lot different very quickly.
Yeah, and I put highlighted, articles, citations from the articles. I've highlighted them in this same turquoise color you see up here in the PowerPoint to help just to direct you, guide you where you could go if you wanted to have further discussions about those studies. Of course, you can read the other studies that are not highlighted. It's just that those that are highlighted were ones that I am interested in pertaining to my work. Some of the genes in there I have worked with myself. And so if you had questions from a person that you can reach out to, I'm right here. I can pop a stream up and we can talk about that specific study, or the methods of that study. How did they do this and how did they do that? And we can go through it step by step. That's why I was highlighting some studies in there. But it's important to read some of those studies. You're not going to get through all of them. I didn't get through all of them, but it's important to read the way they talk about their work and the different people that they're referencing, and look at how far back they reference different studies, and what their goal and objective was. What were they trying to do with this study? Like read the abstract and then read the conclusion statement at the end, and then try to look through the methods and the results and see, okay, did they accomplish their goals? Sometimes they don't. Sometimes they don't accomplish their goal, which is why I'm being completely transparent and showing you on some of these studies, their figures. Look through their figures. The way they word things, the nomenclature, some of the jargon is a little, whatever. But I can help you with that. I'm here as a resource. Dr. Dunbar is here as a resource. BGS, many of us here that work in science, and Michael and the Super Tree is there in medicine, and Rodney, they're in medicine. So there's a lot of these words and concentrations and stuff. It absolutely makes sense to them, and for myself. But if it doesn't make sense to you, like we were talking earlier, me, Gold Professor, and myself, we're talking about some things like, you have to talk this stuff out. And in that, you can reduce your stress. I've learned. Absolutely. In that, you can reduce your stress. Like just knowing a little bit more helps a lot.
Yeah, and if anyone has any questions for me, I put my email address at the beginning and the end of all my videos.
So. Yes, you do. I have the link, and if you're not sure where his videos are, I have the link to that video in the description box. So then you would be able to see his email.
I mean, I don't know what Nicole knows, but
what are you talking about? What do you mean? Huh?
Look, you got the title of Science Queen. Don't even front, Nicole.
I know, man.
That's all I can do. I just do what I can, y'all. I mean, it's hard.
You know what though, Nicole? Like, how many people can say they know a geneticist, knowing that in this country, that field by itself that you're occupied in has launched so many philosophical weapons against our demographic? It's not even funny. Mm-hmm. You see what I'm saying? Like, it really is a profound thing, and it really is a unique thing we can say we have access to in the sphere. And we can't thank you enough, because you could be charging so much for this that it'd be way outside of our ability to really utilize it.
I could, and a lot of my colleagues do charge, and that's their business, but they do a lot more with anytime we have discussions, and the first thing they say is, well, did you patent it? Do you have a copyright? Right. Did you go through FDA? Blah, blah, blah. And I'm like, look, fool, I got GLP and GMP. Don't come at me. Like, I got my stuff together. Do you have your stuff together? We come at each other back and forth. They're like, I'm about to go talk to VCs and do my, venture capitalists, do my thing, da, da, da. And I'm like, okay, you ain't in the lab, fool. Like, you ain't doing no work. Like, that thing, you can go talk to who you want to talk to. You can make all the money doing everything. If you're not helping people, if you're not teaching people, how to look at this stuff, don't just gloss over these studies. Go through the study, look for the details, search for keywords, search for the words, like look it up. Like that's what you're supposed to do as a scientist. Somebody taught you, you didn't know nothing, and they taught you from scratch. You didn't know how to pick up a pipette. You were scared of it. Like you didn't know what a P1000 was, a pipette that maximum is 1,000 microliters. You didn't know what that was. You were like, what? Like, you didn't know what it was. And they had you taking the trash out. So don't pretend like you started and you just knew everything. And you talking to, you talking to VCs now and talking to the FDA and all these alphabet boys. at the end of the day, if you ain't teaching nobody, then why are you even here? Like, why are you even doing the science? If you're not showing anybody the way for them to learn, then why are you doing it? Like, what's the point?
Yeah, Nicole. And the other thing is, like, in our community, there's a distinct need. I mean, here in DC, I mean, there's so many. It's a government city, but I mean, so many people who tell me you're the first scientist I ever met. You know, you're the first Black scientist. You know, so it's just there's a need.
There absolutely is, and I think there needs to be the 3 levels. You know, in ninjutsu, we learned that you've got the higher level teaching, the middle level teaching, and then the lower level teaching, which really stems from teaching the theory and concept so that you can relate to it in an overarching sense, then showing the scientific method and how it can be applied, and then demonstrating the practical level that you see it in your everyday life. You know, maybe there's a philosophy with that, you know, in the sciences, but that's how we teach it in the general martial arts. And we're missing a lot of those different levels of transition. And, you know, let's be honest, the overwhelming majority of us that do get into those fields don't ever have an opportunity to really bring this back to the demographic that they came from, so that, you know, this information can be retained and we can inspire other people to want to be concerned. You know, we've got to start at that real ground-based level to be able to communicate why these things are important to us and how they affect you immediately. And number 2, demonstrate why policy is important, because then you can start getting resources from the government that it's already spending money on, so that you can redirect that to places you live at.
Yeah. You know, because we, for instance, we were talking about the mosquitoes earlier. I know Professor had to step out, and that's cool, but we were talking about that earlier, you know, and he'll listen to the later, you know, talking about mosquitoes. Maybe I live in Florida. I live in Miami. We're doing these mosquitoes. Okay, well, I don't really want that. You need to, not knowing that they're going to be genetically modifying these mosquitoes in your area for whatever reason, you need to go to the city council. You can take those articles with you. Like, this is what they're doing. This is the downside. This article talks about limitations to this genetically modified organism. I don't want this genetically modified organism here. And I know a lot of people didn't go because they didn't know. They didn't know they could, you know, they could challenge. This is research. You have research, and then you have politics. You know, they should be separate. They're not always separate, but I'm just saying, but they should be separate. And you can go and say, hey, you know, I don't really want these types of mosquitoes released. Have they been released? How do they affect our food supply? How do they affect this? How do they affect the water or the environment? In Florida, they just said, you know, forget all that. We're just go ahead and in real life, they already were like, well, we just go ahead and release them.
Right.
You know, 21. The people were like, why? Why are you doing this? But the thing is, you're not empowered when you show up as a citizen and all you can say is why. Why are you going to do that? That's wrong. What about our kids? They don't care about your kids, clearly. Fact. But if you could say this research shows that this type of genetically modified organism is a detriment to this particular species of organism or this particular habitat, we do not want this here. You know, who is behind this? Who is doing this? Like, you know, getting that information together.
Yeah, that stuff passes damn near uncontested. And there's almost always financial incentive behind it because freaking CC Corp shows up like, guys, we got this experimental thing and we got all this government funding to go ahead and do it. We're gonna help the mosquitoes. You don't like mosquitoes, right? And that's how this crap goes through.
Right. So, they show all the diseases that have been you know, all these diseases that are coming from mosquitoes and all this and all that. I'm like, wait. So your solution to that, question their mentality. So your solution to all the mosquitoes is let's create a species of mosquitoes, whole new species that costs a lot of money to do. We're going to do all that so that over so many hundreds of generations, they'll be gone. That's your solution. That's what you, how about you just make more frogs to eat the mosquito? Like, come on, man. What are you talking about? Are we seriously sitting here having, this is what, if I were a citizen at the time in that area, the type of conversation I'll be having. Are you serious about that, dude? Are you really going to do that? Does that make sense? Wouldn't it be cheaper just to, I don't know, buy some frogs and throw them out there? Like, you know.
Let's see who makes money off of that. We go back to the incentive. And so like, this is how we got things like the prison industrial complex off the strength of the 94 crime bill. You know, we could have had our social scientists show up like, look, you're going to create a financial incentive, which creates a conflict of interest with the state to make that happen if you do so, which literally could have derailed that whole entire dynamic being put forward, because it would have been made less lucrative for these states to come in and, one, use the additional funding that these counties are getting to have these prisons there, and then two, allowing those citizens to count in the electoral college weight for its citizen count. So
Right.
here you are messing around with that county's ability to extract even more resources, taking them away from the locations of other places in the state that would need it most. Like, if we don't understand that nothing dealing with human behavior or in nature happens in a vacuum, we're going to fail to realize how you as an individual are not indicative to what's going on with the whole. And if you want to affect your immediate surrounding where you live in, you need to understand the many overlapping spheres of influence that participate in the creation of your environment. And that includes the social environment. So it's why things like this are so critical to talk about, so that we can have a level of understanding to understand that, look, if you don't take advantage of this, somebody else is, and they've made it a practice to not give a daggone about how you feel once you're at a certain level of melanin. That's all I'm gonna say to that.
I'm gonna give a thank you for that. I'm gonna give a shout out to India XOXO, hugs and kisses. She said, "I'm so happy I was able to listen to this live stream. Thank you, Nicole. Thank you very much, Miss India, for showing up. Everyone coming, even if you come later and watch the replay, I appreciate you. Much respect and thank you to everybody that came. I'm about to shut this down. I gotta do some reading on this G6PD, and yeah." I was reading Dr. Dunbar. I was reading the other night, and I was like, "Oh man, I gotta take notes." I was sitting here trying to take notes. Thank you, West Coast African, for that Cash App. I appreciate you.
Were you reading that paper, or did you go find some more stuff?
I found some more stuff, but I was reading. I found stuff as I read. I go and find other things. You know, I look at the references and take notes of that paper, and then references of the of that paper. Like, I take notes under that. So I was taking notes from like several different sources, and man, you know, they have so many things going on with this deficiency, and I decided to transition my notes from my notebook into PowerPoint. So I got a PowerPoint, you know, with a few slides, not too many slides yet.
Okay.
Adding some slides to it. So that will help me prepare for a discussion with you and other people. Like, you know, if we're talking about G6PD, that will help me. Like, when I'm looking at your videos again, that helps me to understand better. Like, I do that with a lot of topics. I just say, man, forget it. I'm sitting here watching, you know, Raised by Wolves, and I'm like, let me just make a PowerPoint. Let me just start this PowerPoint. You know.
Okay. Well, I have to do some more reading myself on that.
Yeah. And I wanted to know, I have questions. One of them is on the G6PD. I'm still trying to find some stuff on the aging, you know, aging of that, like how aging is contributing or may or may not contribute to the deficiency, or, you know, like aggressive stages of the deficiency. So I'm trying to read through that as well. But yeah, I appreciate y'all coming out. This is cool. I know some people said they're gonna have to watch it from the beginning. Okay, y'all.
you know, Nicole, before we go, I'm gonna say this. You know, the cool thing about the G6PD is that I stumbled upon that by accident.
Yeah.
I was shooting a video on hydroxychloroquine and chloroquine, and someone came in, and that's how that whole thing started. So sometimes you find things by accident.
Yeah. Yeah, that's sometimes the best thing to find is something that you find by accident, because you are so objective. You weren't looking for it already, so you have to go in with open eyes and open ears and try to see everything you can.
That's how I came across social dominance theory.
Yeah, I can see that.
Oh, Nicole, by the way, speaking of theories, you know, someone just came under one of my videos. Oh, I think it was my 5G video. And I forgot I uploaded that. That was back in March. Someone asked me about terrain theory.
Oh, Lord.
Wow.
Oh, Lord.
More flat earth arguments.
Well, terrain theory, solution for that, you just go watch Nameless Protagonist IG and get all his recipes. Steal all his recipes and everything. He has all his I was like, oh dang, that look good. I was like, what? Look at this, look at that. I was like, oh, that look good. I'm sitting here like, oh my gosh, I know I can't make that. I'll try. I'll try to do it. But yeah, that's what I would tell them to do. Just go look at some healthy foods and stuff like that. Terrain theory, I can't. I'm done with that. Everything I'm reading, I'm like, man, come on.
Well, this is the first place I heard about terrain theories on your channel.
Yeah, I know. I feel like, okay, man, nah, it is what it is. May not be the last, but it may not be the last time we hear it. But I don't know. I'm still trying to figure it out. I mean, when the dude that I'm hearing, when I'm looking at the videos about it, and then your eyes big and wide, like I can't. I'm like, who is this guy right here?
Like,
and I'm asking people who are in different fields, of different philosophy, understand different philosophies and stuff, and they're like, what? These are older people. I'm like, if anybody knows, this older person knows, because they teach me about certain philosophical ideas and stuff. And I'm like, what is this? And they're like, that sounds like a hoax. Like, that sounds like some kind of scam. I was like, okay. And, they would tell me, like, I've seen a lot of scams in my time. That looks like a scam right there. One of them said it looked like a, he said it looked like one of those old hippie types of thing they're trying to pull on people again. Oh Lord, I thought they were all gone. He said, I thought they all got jobs. I was like, oh God.
What do you think happens when you introduce these standards of learning, around year 2000, and everybody's teaching a test and nobody's really getting into the subjects anymore? They just have these sequenced, memorized answers that don't have any context associated with them. So, I mean, you get questions that don't get asked. Like, one of the main things in the sphere that's going around now, for example, is people's failure to recognize that a human being's decision-making process and a demographics decision-making process do have a correspondence to each other, but they're not a hard correspondence. The same way you would identify elements in the periodic table and understand that when you aggregate those elements, they behave a certain type of way. Like, your individual behavior can be different than the group, but the group's behavior in general, on average, can affect the way you make your decisions.
Of course.
And this is what creates social psychology. But we've got a whole full-on disregard of that going on into the sector. Going, well, you just should have known through osmosis because reasons. Like, how do you constructively solve issues, with that kind of logic? But
you can't. That's the point. They don't want to solve the issues. That's the point. If you don't want to produce or develop some type of solution to the problem, then you're going to always have conversations and arguments like that. You don't want to solve the problem. Some people, I think some Negroes, we like to, we're afraid of, we know the solution, but we're afraid to implement it, because then it puts you're accountable. You have to implement it. You have to finish it all the way through. Even if it doesn't work, you try it. You try to get this thing off the ground, and somebody saw you trying, and maybe they'll do it better next time. But you don't even want to do that. Like, come on, and then you want to blame people for stuff. At least they tried, even if they didn't get as far. You're like, well, they should have went. They should have got 100 miles. They got 98 miles. They should have went 100.
Well, I did. I got 100 miles.
They failed.
I got it. What's the problem? I don't understand what the problem is. I did it. Like, damn.
Yeah, or the other way, right? I did it. Why you can't do it? Well, you had everything to your advantage, apparently. Or you maybe you had a different. You came from a different environment. You had more support in your environment. Everybody doesn't have that same environment. Well, why not? Okay.
Right. There we go. Well, they made the choice, right? I made the choice.
I made my choice. It's like, and women do it, men do it, women do it. I hear women saying, like, I made my choice. Why you ain't no better than me? And blah, blah, blah. I did this, I did that. You should have did it too. I'm like, I didn't want to do that. I wanted to do this over here. And if I'm doing something that's more difficult to execute, then it's going to take longer for me to study for it and get prepared for the assessments, the tests. It's going to take longer for me to get any kind of, acknowledgement, which I'm not looking for. But it's going to take longer for me to get to that point where I'm admired as a person of a lot of knowledge. It's going to take longer. I'm not looking for that. I'm just, tirelessly going through the motions and working, getting better and improving my craft. But, you can't criticize me like, oh, well, it shouldn't have taken you that long to do X, Y, Z. What are you doing right now?
Right. So I wanted to highlight the point that at the end of the day, just because you graduated, just because you've got a credential, doesn't mean you're done talking about what you just went to school for, correct?
Facts.
Okay. I was just making sure because, I don't got no credentials. So, people be thinking like, this dude don't know what I'm talking about. I made it. Like, okay. Okay.
I feel like, it's like, okay, credentials like that. Yeah. That ain't even it. Because I know plenty of people that got all kinds of degrees and is dumb as a rock. They don't even know what's going on in the world. And they still out here talking about, they got degrees talking about 5G is the enemy of everybody. It's the human enemy and all this. They probably talking about terrain and, theory and all that, and talking about how bleach do work. It does work. Why but you can't you can't ingest that, dog. Like you
guess
can't.
What else it destroys?
Well, I'm just gonna inhale it. Okay, you're gonna die. Like you're gonna die. You're not smart. Like, what are you doing? What are you doing? And they're like, no, I'm smart. I got all my degrees on the wall. Like, okay, well, it might as well be Legos on the wall because it doesn't mean anything. Like that doesn't mean you need to have like the common sense and the basic understanding of like what is right and what is wrong before you can get to telling me about what I'm supposed to do with my house when it comes to coronavirus. You can't tell me nothing. Like, you need to sit down. And that's why I have a lot of issues. Like, I really wish wherever BGS is in the universe, wherever he's floating around somewhere in the clouds, him and then like Dr. Dunbar got him coming to his channel. Different people that have these crazy outlandish ideas, come when we're live, and holler at us, be like, hey, Nikole, nameless, Dr. Dunbar, I think that in my equivocation and my intellectual inspiration, I think that through motivation and elaboration, I will come to you and give you a quick synopsis and hypothesis of what I have developed. Now, I think that 5G causes corona. So be like, dude, why you went through all of that?
Well, I love it. I love it when BGS takes the person through the process of elimination, question by question. Right. And then finally he just dropped everybody else down.
Be like, okay, Dr. Dunbar, through the process of elimination, I will continue my elaboration. What was the dude on Live in Color? Oh my God, I might play that one time.
Oh, yeah.
Yeah, and he kept using all them big words that were totally out of context, but trying to rhyme them and stuff. But yeah, you're right. As soon as he does the process of elimination, and he's like, okay, bye. Like, all right, bye, dude, bye.
But it's funny because sometimes people don't know that they've already checkmated themselves.
They don't know, yeah.
And they just keep going, and he has to walk them back, and walk them back, and walk them back, and they swear they right.
Yeah.
I think that's part of that conditioning of having the arguments with, their mother and then the baby mama with this kind of stuff back and forth. So people, again, I don't know what it is. We just have an issue with seeing the bigger picture and failing to realize that, there are correspondences and correlations, and we need to take advantage of different perspectives so that we can come up to a solution, because that's what it should be at the end of the day. The solution, not being right.
And that's what I'm saying. Like, everything I have and pertains to, the science and technology, like this is what they have said for this particular project or whatever they're doing or event or something. And I'm just giving, giving a subjective perspective of it, just giving my view, but I'm basing it in what they've said. Like, you can see what they did here. You can see that's their data. I didn't make the data up. I don't have that ability to make the data up from 2003, 2005, 2008. You can see that the data, the experiments is getting more and more, not only improved technology, but it's aggressive forms of technology and engineering as well, that's going into that. Technology meaning the hardware, but the engineering meaning the thought that goes into putting these different things together. So that's all I'm saying. Like, all I can do is show you this is what they did. Why? This is what they said. This is why they said they did it. And you can take it how you want to take it. Is it relevant to what you're doing, what we're dealing with now? Yes. Looking at that timeline, definitely. I just hope that everybody can get out of your own little bubble, opinion bubble that you have, and try to think outside the box. If you don't mind, if you could for a moment, that would be great. And I like doing that. I mean, I think outside the box all the time. I'm listening to Dr. Dunbar and some of the stuff with the pharmacology. I was like, man, he making me I gotta go read. God dang, gotta read again. And I go get my little drug book. And I'm looking through there and looking through my chemistry book. I haven't opened those books in a long, long time. I gotta go look through those books. I was like, man,
fam, it's humbling. Y'all teach me.
Learning from him and a lot of other people out here that are Peak Prosperity and BGS and even Nameless, like other people that are doing different subject matter. I'm like, it is very humbling.
Well, Nicole, I have to relearn some of the molecular biology and you talk about some of those things are fuzzy for me.
Yeah.
But they serve as a foundation to kind of understand why certain things are talked about the way they are. We have to just be sort of honest. Like, we've got a huge comprehension disparity when it comes to being able to relate to some of these topics, especially if they get more into STEM or in the abstract. And that's not being said as an insult, but it's being said as a, basis level of coverage, because we can get into our adulthood without needing to know this stuff. So, the reality is that environment is changing. People are being left to educate themselves on their own volition now. So we're slowly but surely going back to the village model. And I think that village model is being adapted online, and it's bringing people who normally would never come across each other into direct vicinity. So in a sense, we're going back to school again just through proxy. So I think this is something we can kind of take advantage of. I mean, could you imagine if we got the PhDs in the sector and just kind of like had an online summit of, PhDs in the Black community, and then break it down by subject and, just make a general state of the manosphere sort of deal. And we talk about some of the issues that, either may be related to COVID, may be related to current events, and suggestions that could help push things forward in the future. Like, that could be an amazing source of useful information that could be archived and saved for later and serve as a basis for content in the future. Not to tell what people should do, but even like second and third tier content creators like myself that are, just walking in, let's break down what the PhDs were talking about. Let's break down what some of the topics that were brought up during the last meeting. You know what I'm saying? And that can help propagate and solidify some of these concepts, which is naturally going to flow back to people's families. And, being better informed will make having the conversation about power and politics and going in that direction a lot more real and relatable and less fuzzy and far more removed from the gynocratic conditioning folks are used to, It'd
be a lot easier. I agree. I was, they had I spoke about this conference. They had a FBA conference a couple of weeks ago, and I was like, oh, they put so many people at risk, for COVID there in Atlanta. And then I thought about that about, I guess, maybe 3 or 4 days ago. It was earlier this week, and I was thinking, we should have like a conference or something, but online, of course. And so what you're saying, I'm sure many people have thought of as well. And in order to do it, I think Tori had mentioned it one time. He was like, what if we all met up somewhere? We had like a conference or meeting or whatever. And that it was a good suggestion, but I just don't know how it went, if it went anywhere. But it would be easier here, the way we are now,
Right.
having different topics, having different things. That's a good idea.
Like a Zoom conference or something, and
right.
sit back.
Yeah, and everybody comes and they give their presentations and have question and answer. I mean, the way our conferences are set up, well, now they're all digital, but when we had in-person conferences, and one of them was over 40,000 people that would come to that conference every year. It's annual in a different city. And it was like a 4-day conference, but most people were there for like 6 days, like a whole week, basically. And the 4 days they had content, it was from 7 a.m. until 6 p.m. every day. And there were so many things going on. I mean, to accommodate 40,000 people, you have to have like 100 things going on at once. And that's literally what was happening. So you could never get to everything, but they had this booklet that was really thick. And that was what people followed as they wanted to choose where they wanted to go. We had to present posters or oral presentations. And the oral presentation was presented in front of like 50 people. In a smaller room, and your poster is unlimited number of people that'll come by and talk to you, but you had everything in order. You had to have a presentation, a speech for your work. And it wasn't just my area, my discipline of the genetics and engineering portion. You have physicians there presenting case studies, presenting results from their clinical trials. You had nurses there presenting new preventive strategies that they use in nursing. It was all types of things. You had the vendors there from different science and technology companies that we buy stuff from all the time. And I'm up there trying to get stuff for free. Anyways, so I'm trying to get discounts and my little vouchers and stuff. Let me get the hookup, man. Let me get the hookup. Come on, man. Let me get that hookup, man. I know you got 20% off. Let me get 30% off. I'm trying to get money. I'm trying to save money for my lab, man. Let me get that 70% off, man. And they would give it to you. They write you a script, write you a little, put your barcode on there and stamp it. I'm like, oh, so next time you order, you get 70% off, up to a certain amount. I didn't catch that. Up to a certain amount you're ordering, right? You can't order $1,000 worth of stuff and get 70% off. No. But it was still a discount, and making business connections with people, exchanging your business card, all of that, that would be really good. I'm glad you suggested that here. And, keep suggesting that, keep that in the atmosphere. That's a good thing.
Yeah, I think that's what I'll start doing, because, this would also serve as kind of to stay on topic, an antibody towards dissenting ideas that the sphere only wants to sit around and talk about the quote same old things, which,
right.
I'd like for them to then point out how many opportunities they get to take people with all of these credentials to sit around and present solution-based status reports. That's useful information people pay for. So, I mean,
oh, yeah, they would have to pay, because there would be no other way to keep trolls out. You can't avoid the trolls. You'd have to pay for some type of access code or access link or whatever that's only for each IP address. You'd have to do it that way, because trolls and anybody that's a detractor, they're gonna try to come in, try to come in, and yelling in the chat in all caps, "Get your hand out of my pocket," in all caps and all that kind of crazy stuff. We don't have time for that. When BGS is trying to talk about something, geopolitics, and you in here talking, yelling in the chat, get out of here,
Right. Yeah, so we definitely should be able to probably sit down and throw some emails out and get a feeler to see who would be interested and who may not be. Like, even if we get 2 or 3 people, that in and of itself would still be a benefit to at least, talk about certain things. I love listening to anything you guys always have to offer. I always learn something new, and it gives me new ways to look at information I already know and links to go and add to that portfolio. So it's how we kind of do that back loop to feed each other.
Yeah, I'm about to hop off here. I got people emailing me about my samples. I don't know what is going on.
Well, I was gonna say before we go, at the end of the thing, we could have a BGS award.
Oh yeah.
No arguments there.
I know, right?
None. Zero resistance. I co-sign.
So.
Oh, thank you. I have another.
Well, thanks for having us up.
J.A. Rogers, thank you for the Cash App. It says fantastic live stream. Only joined at the 2-hour mark. When I saw the word fantastic, I was thinking something else anyway. Like y'all already know, but anyway, because we don't really. I'll talk about that on another stream. Anyway, I appreciate y'all coming. This was so awesome.
Absolutely.
I want to come back later on this evening. We'll see. I really do. I want to talk about something totally different. I want to get people's feel of the show Lovecraft Country, and hopefully everybody has seen things by now and watch Dr. T.S. Johnson give his commentary on that. So yeah,
most definitely.
Oh, thank you, Sphinx man. Thank you for that.
One day, Nicole, we should do. I got a few things I need to show you. I've been doing some math, literally constructing a nice little formula to kind of address social dominance theory and how it can apply to a term I'm trying to dub demographical inertia. So it's pretty interesting, but we'll see about that. And then the conversation on epigenetics and behavior and the implications that social dominance theory could be having on epigenetics.
Epigenetics, okay.
In other words, conditioned behavior to create, cultural dispositions that may be getting passed on a genetic level through trauma.
I'm writing it down.
Yeah, there's been a lot of cases, particularly folks that came out of World War II. So some interesting models to follow after. So slavery would definitely make a strong comparative and candidate to that.
Yes, I know Angry Man has mentioned that a couple of times, and the epigenetics with slavery. Yeah, and I literally said like there would not be any major studies on that in the past because if you notice that there are epigenetic changes that are occurring due to the traumatic effects of slavery and residual effects of slavery, then that would be considered a health condition. That would be considered there's a health aspect to that.
Yep.
This is an existing condition. This is almost a syndrome or an illness that, you could roll that into a lot of different things.
Which is why I wanted to bring that up in conjunction with your topic of today, because, there was the implication of how pre-existing conditions could play a role into your susceptibility, not only with genetic markers with regards to COVID and all of that, but, we talked about the stress hormones, i.e. the cortisol levels being higher, the high blood pressure scenario, and how a lot of these things may be not only encouraged by the culture, but again, there could be genetic implications of that. So it's just putting another higher power microscope at the algorithm, you know what I'm saying, to see the points of connection. So thought maybe you may be interested.
I just wrote it down. I put TMP, epigenetics, and social dominance theory on my notes. I put nucleus male, mitochondria female at the top, TMP. Every time my notes is on there, that I hadn't thought of with the nucleus and mitochondria, but that's something that would definitely be interesting to discuss, from an esoteric level. Yeah.
We already know where it'd be passed down on then, since everybody has a mother.
Mm-hmm. You're right. I'm not going to talk about nothing more than that. I'm not going to go into that. You're not going to drag me into that. Yes, Cloud. No, you're not. I'm going to get out of here, y'all, because I have to start dinner soon. But I hope you all enjoyed it and that you email me. Let me know, like I said earlier in the stream, let me know if you want a copy of the PowerPoint, and I'll definitely send that to you. Thank you again, Dr. Dunbar, for having me on your stream last Saturday. It was great. And I'm glad that you have made it over 400. You have over 400 subs.
Salute, family.
Appreciate all the help
Huh?
and the support. I appreciate all the help and the support. Appreciate you coming over.
Oh, yeah. You're welcome.
Put a link into your channel again, family. In case
if you can put that in
playback.
Hey,
I
Nicole,
put a
you
link.
I'm driving. Yeah.
Yeah, no problem. I put the link for the last video you did. Of course, that's on your channel. But I'll try to find it. I'm finding it now to share the channel with everybody. So the link I'm putting in right now is Dr. Dunbar's channel. Oops. There we go. And that is Big Discussion 76, Science and Technology by, it's right there, Rashid. His link is right there.
He may be delayed a little bit.
Okay. I forget. Oh, no problem. I know sometimes he is driving and everything, and I hear my art news now, he's like saying that he's driving. So sometimes people can't like look at the banner or it's moving too fast or whatever. So I have that up there. I have his link right there for his channel. If you need anything else from me, y'all know how to hit me up on the email. Once again, thank you for the support. Everybody in the Cash App, the Super Chat, I really appreciate it. Who is showing up late? Chaos? No, I gotta go. I already know you showing up. I'm about to say, who is this coming up here? I'm about to go. Y'all gonna get me in trouble. I have to start dinner now. So I'm gonna holler at y'all later. And y'all be good. Y'all be good. If y'all have anything else y'all want to say, any last words?
Just thanks for having me up. Peace. Doctor, it was good seeing you. Nicole, always a pleasure.
Yeah, same here.
All right, talk to y'all next time.
I'll talk to y'all next time. Be good.